in Cameroon, in University of Yaoundé, and trained as a high school teacher. And his quest for knowledge pushed him to further go beyond high school teaching, and then went got a master's and a PhD in organic synthetic inorganic chemistry. And after that, he has been working in the area of medicinal chemistry as well as synthetic inorganic chemistry. So he has been synthesizing molecules that are of interest for medicinal chemistry. So he is going to be chairing this morning session. And the speaker this morning is Dr. Andres Vasquez. Andres Felipe Vasquez studied first of all in Columbia University of Valley, and then went further to get his master's and PhD from the University of Andes. And from there, he has been focusing from his master's and PhD in computational biology and molecular modeling, and has spent more than 10 years working in this area. Currently, he works at the Institute of Plant Biochemistry in Halle in Germany, and we are very delighted to have him to speak to us this morning. Dr. Vasquez, you have the floor. You have 45 minutes to give us your keynote lecture. Thank you very much.
Thanks a lot, Dr. Kitheka, for a very nice introduction. It's a pleasure for me to be today with you. It's a real honor. So I will start to share my screen. Please confirm that everything is okay. You can hear me, see me, and of course see the slides with no problem. Okay. Can you see my slides with no problem?
It's coming up. Yeah, fine.
Okay, perfect. So we can start. The title of my presentation, as you can see, is focused to spin around how can be drug discovery innovation be greener. Why? But most importantly, is what that means and why it is important. What should pursue that, and why I can put here that it's important to unleash or harness the full potential of PS campaigns and most of it molecular fragmentation. So, especially in terms of how is this seminar organized, I decided to make it or separated in very few chapters, so to say. Most of it related to chemical space PS, some other concepts about molecular fragmentation, natural products, and the potential of the integration. On drug design strategy right now, and finally just with some remarks and perspectives. Just as I was telling you a moment ago, why is this important in drug discovery knowledge? What is happening right now in the beginnings of 2024? Of course, in computational chemistry, there's a lot of things to do in these days. Actually, part of many jobs are, so to say, holy grails in computational chemistry or chemistry in general, including, for example, biocatalysis. I'm looking for, or trying to look for better proteins. Part of them is actually not new to nature in order to make better or faster chemical reactions. Part of other computational chemists are working, for example, in protein folding. Of course, this is really critical in order to try to get a very good understanding about the function of proteins, not only from structure, but also from a sequence. But in my case, I was the topic that was really attractive for me when I was really young, finishing my undergraduate studies and start my first master's degree in Colombia, was trying to understand what is chemical space, what is chemicals multiverse. That is the term that you can see at the beginning of this slide as hidden. The chemical space is in computational chemistry referring to any kind of molecule, to all the set of possible molecules that can be formed by different elements in various organic proportions. Of course, this includes all kinds of organic molecules. In the world of drug discovery, it is spinning around the efficacy in drug development and a specific characteristic in materials. Most of it, for example, from the perspective of chemists, is all about, or most of it, about the potency. But we already know that there is a lot of other important features like added properties or many other kind of important features, including this chemical space and the chemical direction. I'm going to talk a little bit more about it in the next few slides. So understand what is this the meaning of this chemical space of multiverse and find the very grasp concept. We're gonna try to understand what it means in terms of magnitude, in the orders of numbers. I have here just a few examples. For example, Starbucks locations worldwide, a little bit more than 35,000 of them. Cars Tesla's produced up to now is almost 2 million of them. Pennies produced by US Mint, for example, we are just giving a much larger jump in terms of orders of magnitude, about billions or trillions. The stars in the Milky Way galaxy, the more drops on water on Earth, grains of sand on Earth, and we're just dealing with much higher numbers, and drug-like molecules in the chemical space is really big. I cannot put this in my slides, all the zeros that it really means. So definitely we are dealing with a very, very high chemical space, and most of it a world of possibilities. But in the next few slides, we are going to return to this point in order to understand another question of interest, that is how much of this space we already know, and why it is important to try to navigate through it faster and more effectively in terms of many other resources apart from time. Many pioneering patents have been done in the last decade, I would say, in order to characterize the chemicals. The first larger theoretical chemistry journal paper about or trying to characterize this chemical space was published in 2013. And the idea was to try to understand what it is located in terms of rule of 5, the Biosynthetic rule, very classical for drug design and discovery, but in terms, for example, in a general perspective from chemistry, where we're dealing about heterocyclic, carbocyclic, or heteroaromatic groups, and many others. Well, what does it look like? This the chemical we already know. In order to try to pursue all these ideas in a more effective way. Sorry, something happened. I heard some, or it's just people entering the room. Doctor Kitheka? Can you hear me?
Hear you very well.
Okay,
everything looks fine. Your slides are moving.
Oh, okay. I will just tell you that in order to get to answer in terms of chemical space and the navigation, effective navigation of it, one of the main problems is about what is the cost, the timeline, and the complexity of this kind of procedures. Of course, in terms of computational biology and computational chemistry, there are most of it related to the early parts of this entire pipeline. Most of it target identification, function study, and drug design and optimization. So there's a lot of very nice capabilities or strengths that is important to go this way. Most of it using virtual screening in order to make faster possibilities of the navigation of the clinical space in any kind of drug discovery campaign. For example, in our group, in the IPB, Computational Chemistry group, within the bioorganic chemistry department, here in Halle, in Germany, we have, encompassed a lot of different kind of strategies and approaches and try to construct a very solid pipeline. As you can see in the middle, in the middle part of my slides, is try to put all of these kind of approaches in a very robust workflow. There is most of it related to the virtual screen. Usually, in a drug discovery campaign, you start with millions, billions, or as you could see just a moment ago, many kind of compounds. And it's impossible for, in terms of cost, time, and human effort, and infrastructure, actually trying to deal with so many compounds, to test it and get it to experimental scenarios. So definitely, the idea is to try to deal with most of the noise, so to say, along this process, and get only the compound that looks more promising to continue and to follow up studies in the near future. Compared with the classical high throughput screening, just fully experimental, the virtual screening can provide a much lower cost to build and maintain library of compounds, and even to the possibility to investigate compounds not synthesized yet, which is of course not possible in an experimental scenario. So definitely the idea in virtual screening is to complement this kind of procedures as well as possible. Many classical methods have been employed during decades for BS campaigns since the early or mid-80s up to now. We are talking about more or less 4 decades, I would say. Part of, or one of the most famous is molecular docking. I am pretty sure that you have seen a lot of this kind of applications from these approaches during many other seminars and conferences in this event. I don't want to go into much details about the technique and how it functions. Another approach that can be complemented some way with molecular docking is pharmacophore modeling. That, as you can see in the image, is trying to get a 3D abstraction. But what are the molecular features important for a ligand in order to assure or improve their interaction potential with the biomolecule of interest that is usually protein. So when I was starting my first master's degree, I would say this was about 2010. I started to detect many research works at that time that were trying, on the one hand, try to compare both kind of techniques, but curiously, part of this were focused only in the possibility of combining both of them in order to try to make a cross-check of the pose and interaction, and in the case of molecular docking, get predicted binding energy and what is happening. Of course, after all these comparisons and trying to understand if I use both of them, why to use one before the other, the usual trend is to use pharmacophore modeling before molecular docking, because it's much faster and is more effective to deal with flexibility in the protein, for example, if it's the molecular receptor in this case, compared to molecular docking that usually is more complex and time-consuming and in-consuming actually in terms of processing time and computational infrastructure. So At that time, I was coming to you, I was starting my first master's degree, for example, I can share with you a very nice example of what is important in a BS campaign or visual screening. At that time, I was working on human DHFR inhibitors. They're very important, for example, in cancer, but I understand this same protein is important in many other perspectives, even, for example, in anti-malaria compounds. But at that time, I was trying to understand what it was important in order to compare classical compounds that as methotrexate or palatrexate compared with non-classical. This differential is really critical in this case because, as you can see, part of the molecule is most of it maintained, this diaminopyrimidine group, but the rest of the molecule, especially this glutamate tail, is completely replaced by a more lipophilic non-classical group. This is critical because we already know that glutamate tail is really important not only to bind to the final target, in this case the enzyme, the HFR, but it's also important in order to get interaction with transports in order to get through the cell, specifically and interact with the final target, with the final enzymes, but actually interact with with some other enzymes, able to detect and recognize this glutamate and polymerize the compound. This has an implications for the time is inside the cell, this kind of molecules, but I don't want to go into more details. The take-home message in this case is trying to understand that I was pursuing some kind of novel compounds that maintaining a specific chemical scaffold of interest. That it wasn't. I understand that it was really critical in order to try to maintain a specific H bonds that you can see here, comparing in the methotrexate that was my positive control, my test compounds in B and C squares, and compared with a negative control that I use you can see in square number B, completely different of interaction that I was expecting in the comparison with the other three. After checking these compounds in human cells and in mouse cells, specifically melanoma, we could find that both of them were promising. Both of it, the compound number C2. Why I mention all of this anecdote from previous experiments? You remember that I mentioned the aminopyrimidine, that was the region I want to maintain, no change, but the BR region or variable region, I want to get rid of this glutamate and try to pursue a more lipophilic compound. That was important in order to get get rid of the necessity of this interact with this transport first, get through the cell, and interact finally with my enzyme target. It was important to get through the cell to pass its diffusion. So at that time, I mean, about 10 or 11 years ago, there was published a very nice seminal work in a Journal of Clinical Information and Modeling by Sayer and colleagues that recognizes some details in vitro screening. And the most important part of it is that the potency is not the only objective to pursue in a virtual screening campaign, but actually it could be not the most important of it. So what it means that in my case I had to sacrifice part of the potency offered by this glutamate in order to get a lipophilic compound, not so or lipophilic chemical moiety, not so potent, but I was trying to deal with some persistent associated mechanisms. So in a BS strategy, the most important thing is try to start or try to identify many different starting points. That is the hard topic here in order to debate from the basic chemistry. Trying to understand what can be done in terms of this chemical diversity at the very end of any kind of virtual spinning campaign. A very nice technique that has been proof effective and successful in the last, I would say, 15 years or 20 years up to now, is fragment-based drug design. Compared with the classical chemical enumeration, you know that we start with the scaffold and try to decorate it with many kind of, slight modifications and in terms of the R groups and selection of anchoring points around this scaffold. I just took one molecule as an example here. This is glycerin, a natural product. The fragment-based drug design tried to start with the problem in the very beginning with very tiny chemical entities. This has, a number of benefits, including, for example, improving the protein ligand complementarity. You can see here, if you can see my cursor in my slides, explore the chemical space in a more cost-effective way, because we know that we are moving to the size of fragments that is about 180 and 300 Dalton, to give you an idea, and move to the size of complete drug size compounds, so to say. We are dealing about 5,000 Dalton, more or less. So in that area, that range, the growing of the curve is exponential. So that means that in order to explore in a deeper way the chemical space, is technically speaking more effective to deal with fragments compared with drug-sized compounds. Beyond that, there is another kind of nice benefits, including the facilitating of adjustments of balance. For example, in optimization, when you are dealing with dual or multi-target compounds, and the reduction on false negative rate. That is really important, and I mentioned in the previous slide, because the problem with dealing with drug-sized compounds is that, for example, in this case, you could see I have found a very nice scaffold, including the interaction, for example, in a specific area, but part of the molecule has no good interactions or a clash with a steric clash, I mean, and you lose that information eventually. So the idea is not to lose information, use it as good as possible during the complementarity in protein ligand interactions, and finally combine these fragments in order to get a final really good compound. Trying to pursue an idea not only to start with fragments per se, but, for example, dealing with the deconstruction of very large compounds. You can do both things during your BS campaign. You can decide what kind of rules you are going to use in order to cut these molecules, get your fragment library collection, and then you can reconstruct and combine all the groups against all in order to enlarge and expand your own chemical space. Eventually, you can do this with many other open source chemical libraries. Ligand efficiency is a concept really related, intrinsically related with the fragment-based drug design, because it has been proved that when you use fragments and you are starting to normalize the potency of the compound in terms of the size of it, and the size I mean the heavy atoms or non-hydrogen atoms, you are starting to get away from this bias selection to larger ligands. What does this mean? This means that 20 years ago, many researchers on the field that when you are getting to put more and more and more R groups and decorate the scaffolds of many other well-known drugs already known in the literature, you are getting a plateau, or they were getting at that time a plateau in activity. So it means that not necessarily bigger compounds are going to be more potent and eventually more effective. So the idea is to get a very nice potency in terms of not necessarily a larger size, at least not in the very early stages. I mean, hit discovery or hit to lead this discovery steps. That's why the idea behind union efficiency was that kind of important. Mathematically speaking, is really simple, as you can see. Just delta G over N. There is just heavy atoms now. And they have as a kind of criteria for ligand design. What is the product of this kind of case, or what is the opportunity I saw at that time? I mentioned to you there's a very discussed overemphasis on potency in terms of virtual screening campaigns. It's a very important pursuit behind a very nice exploration of the chemical space. But what about fragments that don't necessarily exhibit a high efficiency level during the screening procedure? I mentioned to you, for example, before the example in the variable region of the DHFR inhibitors. Of course, they were not probably so potent at the glutamate tail. I needed to change it, but lipophilic chemical moiety, but it has a very good reason. In my case, was an adjustment of selectivity. But what about chemical innovation? What about how feasible is synthesize this chemical moiety or not? And what is the alerts detected or not in the structural alerts? It's important for ADMET prediction and paints, compounds, this kind of stuff. So why is this important? Actually, many of these or all of these criteria are not mutually exclusive to each other. So if many of them or all of them can happen at the same time, there is a very good reason that I can need to put a fragment as a part of a whole molecule, even if there is no so high diffusion per se. So that's why I started to study what is new in terms of ligand efficiency metrics or indexes, but in the world of fragment radial design. I could find a very nice work. For example, this is Dr. David from a very reputed pharmaceutical in United Kingdom that is called Astex Therapeutics. And this deals with the double delta G over the delta N. What it means that you're starting to grow a very nice and a very potent fragment per se, and eventually you start to decorate and try to compare what kind of R groups and decoration of this initial or seminal scaffold are more promising or not. But even so, this kind of new or newer indexes have some problems. I could detect two of them. On the one hand, it requires a pairwise comparison of compounds that really closely structurally related. And on the other hand, we need to optimization of a highly efficient fragment. So you need to start with a very good fragment, and you need to fully compromise with it in order to continue during the optimization procedures. So what can I do in order to not to deal with these drawbacks? I started to work in 2018, a very nice mathematical formula in trying to understand under the assumption that the ligand efficiency of a full compound can be predicted, can be calculated in terms of the contribution, the relative contribution of any kind of fragment inside the compound. This was theoretically resolved for a linking strategy of fragments, but I need to mention to you that there is some others kind of strategies to combine this kind of fragments in the very end. So yeah, at that time I was working on this basic mathematical formula. That the most important thing to mention here is the possibility of you can get a rescue effect for some kind of fragments. What it means? It means that you could get a very not necessarily a highly potent fragment, but if you had as a companies 2 or 3 or more fragments that are really good, you can save, you can rescue this fragment of interest and take it as a part of the whole compound. You know, this other mathematical term that I called at that time LA delta is a very useful ideal value or transient value in order to try to not to start with a specific fragment of in the very beginning, but you can deal with many kind of fragments at the very beginning. So what it means, practically speaking, that you can go for many comparisons or try to understand all the possible combination of fragments that you need in order to get a very nice compound, even before going beyond in experiments and going beyond many tests in the lab. I will show you very fast what it means in a hypothetical scenario. This is the standard fragment selection. You can see here, for example, my fragment is not above a cutoff value for ligand efficiency according to the standard literature, but I don't know what is happening in the other position. I can put another fragments here in P2 or P3, but I'm not taking into consideration what is happening here, maybe what kind of combination I can do. So just measuring this cutoff value of ligand efficiency for my fragment is just eliminated from any possible combination. What we do at that time, from the on the one hand, try to deal with, okay, maybe I have some other fragments in other positions able to, in general, to rescue my first fragment, and I can, in this way, take it as a part of my final whole compound. On the other hand, if, for example, my combinations, my possible combinations of fragments in the other positions are not so good or not good enough in order to rescue my compound, I can just run a direct elimination of this fragment from the very beginning with no need to go to the lab to synthesize, with no need to go to many other lab experiments for validation of this compound. So definitely it's nice to save a lot of time, human effort, and infrastructure. Yeah. The rescue effect is, at that time I was wondering, is far from being new, or what is happening? I could detect, I'm not going to be in so much detail at this area, but the main idea here is the rescue effect is usually done in later stages of the optimization. So it means that in the decoration of a scaffold during the optimization of a lead compound, many kind of R groups that are not necessarily potent, for example, lipophilic or even cyclic aliphatic, are included. What if I can do this in the very beginning? And that is why this kind of ligand efficiency indexes are really important, and this RGC model and the fragment rescue effect is really important. In order to share with you just very fast some other experiences before with fragment-based drug logic design. This work, for example, I did it during my Ph.D. some years ago. I was working at that time of anti-cancer agents and a part of protein kinases, CDK2 and BGFR2. As you can see here, they are very similar to each other, but I was wondering what is the chance of getting a dual compound able to block this both of these enzymes at the same time, just with one molecule. Able to cover all the area on the binding side. But I had real challenges at that time. Most of it related with what is the the real binding site that I'm pursuing to interact with. Most of these inhibitors are non-inhibitors, one inhibitors, but the two real problems with that is that really toxic because they are really promiscuous. Many other kind of enzymes in the cell use ATP, so any kind of molecule able to use ATP is going to be eventually a problem. And there is a crowded chemical space. So it means that you have eventually really great problems in terms of patentability of this compound, because there is a lot of many kind of modifications of the ATP already synthesized, already tested, and it's really difficult to get something novel in this area. So I could find that a near area in my province was a very hydrophobic pocket near to this big binding site, and this pocket was related to the potential of selectivity. So I tried to pursue that idea and try to do it with fragments in order to construct a compound of interest. I'm going to share some more details about it, but the main idea at this point is that even if fragment-based drug design is promising at that time for getting this recombination, of nice and small chemical scaffolds, it has a very nice, a very problematic, limitation. That if you start with a chemical library of no so good chemical diversity, you are eventually trying to pursue the same kind of model. It's similar to an analogy of a bag of apples. No matter how you cut the apples and you try to paste these apple pieces again, you are going to finalize with apples again, or things that are really similar to apples. So you have to take into mind, okay, maybe fragment-based drug design is okay, or molecular fragmentation is okay, but definitely I need an input library that is really high in terms of chemical diversity. And what can I find this? And in terms of how big is the chemical space that I promised to you, mentioned this order of magnitude at the very beginning of my talk, is that we already know just a tiny part of it, 10 to the power of 26. But the theoretical space is really large. Some authors have talking about, almost 10 to the power of 100. So definitely there is a large gap to be filled, and the chemical diversity is important. Not only the recombination possibilities offered by fragment-based drug design. How can I resolve this? I can explore the chemical space faster and easily. And my answer comes with natural products. There are great sources of chemical diversity, but unfortunately, they have been somehow underexplored. Of course, they have historically been strongly related with drugs, but in terms of how much or how many natural products are visualized and taking part of many chemical libraries, at least open source, are really low. They are most of it. Even if I add all of them right now, around or even below 1 million, compared to drug-like compounds that are in the order of billions of compounds. So definitely there is a large gap in this kind of comparison. But the real, the real opportunity here is to understand not only the MPs are great for chemical diversity, but they are really complex in order to try to synthesize in the lab. They are really complex in order to optimize eventually. But if you combine natural products with fragment-based drug design, you can decrease the complexity level of the natural products compared with giving a very nice input of chemical diversity to the fragment-based drug design. So in this case, we are pursuing a synergistic effect in the combination of both of these kind of nice techniques. Of course, if you combine this in a virtual screening campaign, you're going to do the things much faster in the same scientific project that I mentioned to you when I was a PhD student, I used different kind of natural product libraries, including from some of them from Brazil, from China, and from Africa, in order to get a very nice combination of fragments and try to get use as a filter over two overlapping pharmacophore models, and eventually use these fragments, combined, linked, redocking, and we go to another kind of filtering, eventually with the molecular dynamics too, in order to get a very nice idea of what can be done in terms of new compounds, completely new compounds, in my case, with dual or multi-target capability. This is just part of the results I could get after docking and after molecular dynamics, and the results were very promising. I don't go into details because I am rushing a little bit right now. I don't want to take so much time in this last slide. So the non-extensive fragmentation of MPs was a large part of my PhD at that time. It's a very nice idea because it tries to deal with or to focus on the principle that any kind of molecule, in my case, or my interest in natural products, can be deconstructed. It can be cut in certain parts. But these cuts are usually implemented in literature, including computational literature, in extensive fragments. What does it mean? That if I can detect a potential cutting point here, and here, and you can run this kind of scripts or softwares in the literature or in the web. I mean, you can get immediately all the just the extensive fragments, just cutting all at the same time. So this is not necessarily so good and so informative for a drug discovery scenario, because these fragments, as you could see in the next slide, are really tiny, not so informative, really repetitive, compared with what happened in the information that you can get lost in the intermediate fragments. What happened in the middle between all the possibility combinations in the deconstruction procedure. This kind of intermediate size fragments that I call non-extensive fragments are really important to try to maintain and to try to understand how much has a natural product needs to be deconstructed in order to get very nice compound. I try to use at that time pharmacophore modeling and some kind of test in terms of molecular complexity, molecular skeletons, lipophilicity, and size of these compounds in order to try to understand how promising or not it is pursuing this kind of as I could get as a result, these non-extensive fragments, just as I mentioned a moment ago, are more frequent and less repetitive than the extensive standard fragments. And beyond that, even using pharmacophore modeling, where I could get very nice results in terms of scaffold hopping. That it means that the chemical diversity is not necessarily limited; it can be well maintained. And in terms of even the potential potency, in this case, in the FIT score for pharmacophore modeling, is not only higher than the extensive fragment, but in many cases is pretty much higher than the natural products parent compounds. That what is really nice in order to understand that even in many cases, a natural product by itself is not necessary as a whole. You need maybe to deconstruct a part of it in order to get a very nice fragment of interest. This idea is really nice in order to get or to pursue important taking decision criteria in drug design. If I explain myself, if the level of cutting and deconstruction is too high, maybe you can start just by full synthesis, because then maybe the fragment you need is really tiny, is really small. But on the other hand, if maybe the construction level is not so high, it is possible to deconstruct this part and run to or to pursue eventually a semi-synthetic procedure and a semi, not so deep deconstruction and also deep modification of the parent natural product iron. Just a few slides of most recent research studies or research works. I have the opportunity to participate. This was as a postdoc in Colombia some years ago during the pandemic, in order to try to understand what kind of pharmacophore models can be differentiated between covalent inhibitors for main proteins in SARS-CoV-2 compared to the non-covalent. This is really important in order to pursue, for example, fragment growing and try to pursue an faster optimization of compounds in any category that you are trying to deal with. In literature right now, in this kind of targets for SARS-CoV-2, is really important to mention that even in the literature started to get the first compounds able to be FDA approved against this target. They functioned very good and they were completely well detected by our elucidated models at the very beginning. It was a very large work. We had to deal with at least 70 kind of crystal structures and running 1 or 2 microseconds of molecular dynamic simulations. Combining both things, pharmacophore models and MD simulation. This is called Dynaforce right now in the literature. I want to finalize mentioning the importance not only in a combination of natural products with many other kind of approaches I mentioned to you for my talking, with fragment-based drug design, with virtual screening campaigns, but what can be done in exploration of natural products. We have many mega diverse countries. For example, in my country, Colombia is the most biodiverse country all around the world, but unfortunately there is no still available an open source database, not only for plants, but for fungi, bacteria, and many other kind of organisms that are really important. So with the passing of the time, these kind of bioprospective initiatives are going to take a leading role in order to complement and to push all the chemical space expansion and accelerate the drug discovery innovation, just as my title tried to pursue, screening drug discovery innovation. So just to mention very fast conclusions, the deeper understanding of clinical space is really critical, not only about size mentioned, but also in clinical diversity. Combination of natural products is really nice with fragment-based drug design and visual screening approaches, and the exploration of natural products for many species in many locations, most of them biodiverse, is really critical to pursue as a whole effort. I just want to acknowledge the Institute of Practice, where I'm leader right now as a postdoctoral researcher. Professor Johan Ndarawait, that are my direct bosses, and many other colleagues inside my group and in other departments of the IIT. So I just leaving you with this take-home message that the plethora of small molecules, especially MPs, are hidden gems on the molecular map. But chemoinformatics can serve as our compass to unearth their true therapeutic value. So thank you all. I am remain attentive to any comments or questions.
Thank you very much, Dr. Vasquez. That was quite impressive. I would say that you have been laying down the very foundation, the very blocks for building fragment-based drug design from natural products, which we think is the way forward. Because I also provide a philosophy that compounds that are constructed from natural product fragments hold a lot more potential in terms of bioactivity than the natural products themselves. And we have been talking together with people like Jose Luis Medina Franco, that is going to chair the roundtable discussion this morning, and I'm pretty sure that you have your paths across each other. So, in essence, I was joking yesterday when I was speaking with Professor Rosani da Silva that before the Atlantic came between Africa and Latin America, we were actually neighbors. And the biodiversity should be very similar. So whatever you find in Amazon, you can find in Congo Basin. And so this makes our path to cross-interject, of course. And so I am co-chairing this session with Dr. Evegenii, and if he's there, he can turn on his camera, and he's going to coordinate the question and answer sessions that follows the lecture from Andres. And of course, we take into consideration our break time, our coffee break time. All right, thank you.
Thank you very much. I will remain attentive to any questions and comments.
You are muted.
Yes. Oh, thank you so much. That was brilliant. Thank you so much. Is there any questions?
Hello.
Yeah, Dr. Steven.
Yeah. So that is a very impressive talk, and I think as you brought out what have been our aspirations and what most people have been thinking. Obviously, when you go to the pharmaceutical industry, the complaint is that natural products are very complex. That's why they pursue the kind of voyages that they do, as well as the remedies and that kind of thing. But on this issue of legal definition, I mean, basically, it's how much legal efficiency, just how much potency is produced per carbon or per nitrogen. So how do you reconcile that with the complex or with a variety of natural products that we found from plants or from
This is a very nice question. I would say. I mean, it's really challenging because, of course, behind or intrinsically related to the premise in ligand efficiencies, trying to get a very potent fragment or very potent compound with not necessarily so much atoms. You know, that is important to take into consideration. But on the other hand, I mentioned it's challenging because when you are dealing eventually with natural products, one of the most important features of them is their three-dimensionality. They're not necessarily planar compounds, and this is eventually important even in optimization because it means that you are dealing with so much atoms with sp3, linking points, anchoring points, and this means not necessarily be dealing with so few atoms, so few non-hydrogen atoms. So it means that eventually, with this three-dimensionality, could be some way related. Maybe not in all the cases, but in many cases, could be related with a larger number of atoms per fragment. So we have to be very careful in the long term in order to try to maintain a very healthy balance between how much three-dimensionality and complexity are I trying to pursue, even chemical novelty, starting from natural products, but on the other hand, what kind of ligand efficiency I am trying to pursue. Above or beyond that, the good news is that if you can use this kind of ligand efficiency indexes, just as the index I had the chance to contribute with this RTC model and this LL delta value. The good news is that you can make all the calculation from the very beginning. You don't need to go and to wait from the optimization of a very nice starting natural product or natural product derived compound in order to try to understand, okay, now how can I optimize this kind of compound? No, you can immediately from the very beginning try to understand what I need to combine all the fragments I need in order to pursue a ligand efficiency of interest. So what it means that you can eventually get a fragment not so potent per se, just as I mentioned, but if you have very good companions, you can push it as a whole. So eventually you can pursue both things at the same time, or kill one bear, two bears with the same stone, as you could say in Colombia. So the idea is try to save much more effort from the very beginning. And it means money, because it means time, it means infrastructure, and it means human effort. So there are really critical points, not only for academical work, but for industry. So definitely is something to have in mind and try to consider it and to put it and adapt to your own context, to your own kind of library.
Prof, we have a question from Dr. Chigodlov.
Yeah.
I would like to know if you have carried out any construction of natural product before. What was your experience?
Again, it's a challenging question, because it depends on how can you deconstruct compounds and how you can reconstruct them later. But both of these things are not necessarily the same. But in my case, I have dealt with recap rules. That is one of the most most widely used rules for deconstruct compounds and eventually reconstruct them. But one of the real challenges right now for natural products is that even if it was good for me partially, because I could deconstruct a large part of them, they not necessarily deconstruct anything, or they not necessarily deconstruct to the level I expect. So that it means that this kind of rules have been thinking, have been elucidated in the last, I would say, 20 years or more of it, because recap rules was published in 1998. So I mean that this year is going to be 26 years from the first publication about it. But they were thinking and they were taught, I mean, elucidated for drug-like compounds, not natural products. So definitely there is a lot of challenges in the coming future in order to expand this kind of rules, for example, for natural. And we can do a lot of things about it. Of course, it's not so easy, because we have to deal with many kind of questions about synthetic feasibility, or how easy or how difficult it is to synthesize this kind of compounds, actually in every step. But eventually is going to be theoretically possible to solve this part and try to make it wise for just for natural products specific challenges.
Prof, we have a question from one of the in-person's participants,
yes.
Mr. Metuge. Was there any criteria in selecting and linking the fragments? Any comment about the synthetic accessibility of the proposed compounds compared to the complexity of natural products?
Yes, both of them applies. Synthetic accessibility, that it was the one you mentioned. You can take it into account, not necessarily as a fragment, but you can measure it in your final ranking of full drug size compounds. You can run a very nice pipeline to get an idea of how feasible it is or not. There is several in the web servers, actually free, in the out there, and you can use in order to get this estimation. In my case, I use one of them. It's called Silvia, but I am really sure that there is a lot of or several of them, at least by GitHub, in order to give you a very nice calculation of how feasible are these molecules to synthesize or not. That is one. The other thing you mentioned was about I forgot that part. You mentioned.
Any comment about the synthetic accessibility of the proposal?
Yeah. I remember. Sorry. The combination of the fragments. Yeah. The combination of fragments, technically speaking, it can be through three different ways. One of these is linking, that is just connecting by a linker, different fragments in different, for example, pockets in a if your receptor is a protein. There is one thing that I use, for example, in my RDC model elucidation. But you can do another kind of in literature and most people in industry, they have used a lot the growing strategy. That it means that don't start with all the pockets independently evaluated, most widely used strategies. There is a third one, not so well known, but I used it during my PhD project that I mentioned to you, dealing with dual target compounds. That is a merge strategy. That it means that you have not compounds that are supposed to be linked or one fragment that is supposed to be growing, but you have different compounds that are overlapping each other, and eventually you need to decide what kind of criteria you need to make the combination of this combiner to get a find the whole entire compound. What is the most important reason to pursue this kind of strategy? That when you're dealing with overlapping fragments, you are dealing with intermediate fragments, not so tiny fragments. And that means that this intermediate size fragment, the pose in the binding site is more probable to be maintained as a fragment and compared with the whole compound. It is more difficult to get the same idea when you are dealing with really tiny fragments, because these really tiny fragments can have eventually a completely different pose independently and as part of the whole molecule. So it is really challenging to finalize the idea. So that's why I decided to pursue this merging strategy with very nice results.
Yeah, Prof, we are going to take a last question before the break from Bakari Diallo.
Yeah.
The natural product provide an advantage setting ones in propensity to develop drug resistance.
Sorry, can you repeat me? I didn't understand. Very good question.
Do natural products
Yes.
provide an advantage to setting ones in propensity to develop drug resistance?
Well, again, really challenging. But of course, part of the drug design is dealing with potency, is dealing with ADMET properties, for example, but resistant associated mechanisms is a complete different scenario and a complete different challenge for drug design. But of course, you have to be, to be aware of the importance. Eventually, from my experience, these kind of combinations in the pipeline, not only in terms of natural products, but for example, in terms of fragment-based drug design and visual screening, I am really sure that it's giving you more flexibility as a researcher in order to get very good results eventually. You remember, for example, the DHFR problem that I had when I was a master's student many years ago. I was trying to maintain a specific part of the molecule but change the other. Even if I knew that changes the other part is going to represent for me a sacrifice and a decrease in the potency. And eventually not the problem. But I was trying to gain what? I was trying to get a more probability of overcome or solution or get a solution for drug resistance mechanisms. That in my case were very well known and is very strongly related with melanoma. That has been in my case these two problems. No transport. The transport is mutated or inexistent, and this part of the molecule, the glutamate, needed to pass through. It needed to be recognized this part in order to enter the cell. This was not possible for me at that time. So definitely, I wanted to pursue the possibility of getting a solution for a drug-resistant mechanism. So I'm really sure that this is a very nice example of how to use not only chemical diversity input libraries like natural products, but fragment-based drug design would be really, really good for that.
Thank you so much, Prof, for answering all these questions. It was quite interesting and very, very informative. From there,
Okay, thank you very much, Co-Chair, for that.
We will resume in how many minutes, Prof?
The timetable says that the young investigators session begins at 10 o'clock. That's what the time. Hold on. How sure that this is? 9:45. This is supposed to be at 10 has already passed.
Yes, 7 past 10.
Yeah, let us allow ourselves 5 minutes to refresh, and then we will certainly let's allow ourselves 3 minutes to refresh. We'll take off 10 minutes from each. So most likely the first speaker, we will be the first speaker. They can just allow us to refresh a bit by 3 minutes. Thank you.
Thank you all. Bye.
We're able to bring some fresh air for 3 minutes, actually. When the talks become really, really interesting, sometimes ignore the clock. All right. Thank you very much and welcome back to the Young Investigator session. This session is going to be co-chaired by Dr. Paul Sirek. Paul, are you there? Paul, if you're there, you can turn on your camera. Dr. Jonathan Metuge is also a co-chair. If you're there, you can turn on your camera. Professor James Barr is unfortunately not available. He's on it right now in the Ph.D. phase. And I'm going to take his place as a co-chair of this Young Investigator session. This session is dedicated to young scientists. Young is a relative term, right? Metuge, if you are there, please turn on your camera and I'm going to introduce you. Are you there? You were there a moment ago. Anyway, if you come up, Dr. Metuge, please, we will be willing to introduce you as the co-chair for this Young Investigator session. All right. Cool. So the first speaker of the Young Investigator session, we are very sorry to excuse ourselves for the delay. First speaker is Hannah Augustijn. She's from Wageningen University. Hannah is here. So Hannah, we are very glad that you could honor our invitation. So we are very looking forward for the first session of this workshop. Professor Bidema was always giving us a keynote lecture, and we were very excited by anti-smart, plant-smart, and all the tools that we developed in your group. And Dr. Metuge is here. I would introduce him briefly before introducing Hannah to start talking. So Dr. Metuge is a biochemist by training. For those of you who don't know, he was my high school chemistry teacher. I know if you don't study chemistry in high school, you can never become a professor. Right? So he's someone that can be very challenging, you know. And so, Dr. Metuge, please, a word from you and as a co-chair of this session. We have been following this workshop from the very beginning. So, a word from you, please.
Good morning, everybody. I'm glad we are here. I want to say a big thank you for all the panelists who have given us a brilliant talk on bioinformatics and chemoinformatics. For the students who are going to present, I think I suggest that you send a presentation to Dr. Cheka so that you can mount them in a smooth sequence. Secondly, during the presentation, it would be good that if we have a comment in the forum, then we can read it at the end. So it will be proper that while the presentations are going on, we don't need interventions. I suggest that they allow present, and then we can make contributions at the end. Thank you.
All right. So thank you very much. So Hannah, you can go ahead and take the floor.
Okay, thank you. Share my screen. I didn't send the slides to that one. Do you see my slides now?
Not yet.
Not yet. Oh. Uh-oh. One second. I think I have to do something with the phone. I never used Google Meet.
Oh, you know, I used it before actually. I've received emails, slides from others by email, and I'm now loading them up. I'll just be firing them systematically for others. But you just simply keep your PowerPoint open in another window, and then you can click on slide share, and then of course from slide share you would pick the window that you opened, and then you straight to them. Otherwise, I can just get your slides by email, and I just got support for some people already, and I'm getting ready to.
I'm very sorry.
Yeah, don't mind.
As I don't have permission, but I think I will send you the.
All right. Yeah, the technical team actually was working with permission issues, and we're sure that they're really doing it.
Try one. No. Yeah.
Okay. Yeah.
Could you in the meeting maybe share which email to use if other speakers want to send the presentation to?
Actually, I've just sent an email around.
Oh, it is an email?
So you just check your email and send it back to. Just respond to that email, and I'll get it the moment. Actually, I've just picked up those who have already sent. But sorry for this, and we hope this fixes, because actually we were supposed to have sent it for 24 hours before time according to the program. So it's a pity we are also busy now. But the ones I have been good. I have Hannah's on here, so I'm going to do it straight away. All right. Okay. So here we go. I have Hannah's on here. Perfect.
Yes, thank you. I'm again so sorry for the delay. And I'm very honored to be here. In general, I just heard about the keynotes from Marnix, my supervisor. Unfortunately, he couldn't join this time, so he asked if some of his PhD students would like to present, and yeah, I was really excited about that opportunity, so I feel very honored to talk about my PhD project here, which is on the genomic side of things. So we had a lot of chemoinformatics this morning, and I learned a lot. I'm very impressed. And now I'm going to go into more of the gene clusters, and I hope by the end of this presentation I can have you a little bit excited about the gene clusters and also regulation of those. Next slide, please. Thank you. So why did I go into this direction? So I came across recently this paper where they talked about this novel antibiotic that they discovered, Ndarawait. And this paper impressed me not just because it was amazing work of a lot of people, very, very cool, but what they also described very thoroughly was all the hardships that they went through, all the problems that they had in the end actually getting to the compound. What they also described clearly was that they could find a gene cluster that actually encoded the antibiotic, but this gene cluster was silent in their bacteria. They could not activate it, they could not express it, so they could not get the actual compound, and they put so much work in it to, in the end, get it. So in my work, I really want to dive into that side of things, how to actually activate a cluster of genes encoding the metabolite into production, the regulation of these clusters. Next slide, please. So you may have not heard about Anti-Smash, which is a tool that can detect these clusters of co-localized genes that together encode for a metabolite. But Anti-Smash is really good at finding these patterns in DNA, but up until recently, it wasn't really good at discovering when the compound was being activated. Next, please. Yeah, so this is the main, yes, thank you. This was the main problem. So you have a gene cluster, and very often it's labeled as being unknown in Anti-Smash, and you have no clue what it's actually doing. Next clue. Yeah, but with the use of regulation, if you know how a cluster is being activated, it also can give you some insights in what the function of the BGC is. The BGC is shortened for biosynthetic gene cluster. And what do I mean by this? If, for instance, you have a cluster, you don't know anything, but there is a regulator binding to your gene cluster that is an antibiotic regulator. Then you don't know anything about the cluster of genes, but just by the fact that this antibiotic regulator is binding there, you can actually think, well, maybe this cluster of genes may be related to the production of an antibiotic. Also, it gives you clues on how to express a cluster if there's the same kind of system, but this time it's a regulator related to oxygen, for instance. Maybe if you want to activate a cluster, you don't know how to do that in the lab. If this regulator binds there that's related to oxygen, okay, maybe remove or add oxygen to your experiment to activate this cluster. And all of this can also help you prioritize which clusters may be interested. Like if I think back of the first example of antibiotics, if you're looking for a new antibiotic, wouldn't it make sense to look at all gene clusters that have an antibiotic regulator attached to it to discover something new and narrow down your search into a more specific direction? Next clip. So how do I do this? I look at these patterns in the DNA. These patterns you can see on the left are displayed by a sequence logo, where the height of each position and the letter displayed there represents the occurrences of a certain nucleotide across a variety of experimentally validated binding sites. Very complicated. I make prediction models out of this. I will use the word position mate matrix later on in my presentation. because I converted this into something that I can use to scan the. But this is all very abstract. It's maybe hard to follow, so I will give you an example of how I applied this to actually discover something new. Next slide. So in this presentation, I will talk about a specific type of regulation as an example, which is the regulator DMDR1. Oh, previous one. Can we go get back 2 slides? I'm sorry. Yeah, this one, thank you. So in this system, it's about iron metabolism. So all genes that are related to the uptake of iron are under the control of this specific regulator. It's a very tightly regulated system, as iron is needed for the bacteria, but it's also toxic. So if there's too much, the bacteria will die. So it's a very tight system, which makes it really nice for us to study this regulational element of genes and gene clusters. Next slide. So I looked at this bacteria that's a very well-studied model organism, Streptomyces subicolor, known for its production of a variety of antibiotics. And I looked at this regulator, DMDR1. In the absence of iron, DMDR will actually be lifted and the clusters will get activated. And what I did, I used this position rate matrix that I mentioned before to go through the entire DNA of Streptomyces subicolor and see if I could find a match. And if you do these kind of searches, of course, I can make matches on any kind of certainty. So you need a threshold to say, okay, from this point onwards, I believe that my predictions are going to be real. I used for this the ratio of matches to this sequence logo in coding versus non-coding ratios in the positions in the DNA. So what does that mean? If you have your genes, the regulation is very often upstream of the gene itself. So if they are not upstream, we can say, okay, maybe that hit is not as likely to be one. And that's something that you can see in this plot. So in the upper right corner, almost every of my matches is upstream of the genes. And this is also related to the match score that I have with my position matrix. So the closer I am to a perfect score, the more the ratio of the hits fall in these non-coding upstream of genes. So with using this, I could set a threshold from which onwards I think that my matches might be real. Next one. But the question remains, because you're looking at all these individual matches of these motifs in the DNA, like, are these actually part of like a gene cluster that encodes the metabolite, or are they just somewhere scattered? So for that, we needed another layer of information. Next slide. So for that, I teamed up with a postdoc in my lab, Ndarawait. He worked on this tool called Foko. And Foko is working on correlation in expression systems. So he has expression data. He used, in this case, Streptomyces subicolor under low iron conditions. And he looked at what genes are expressed at that time. And we correlated that with my position matrix scores, the same ones that I showed in the previous slide, to see, is there a match between expression of these genes and these predictions that I made on where these regulators are actually binding in the DNA. If you click one more time. Thank you. You can see, if we set some thresholds on this correlation plot, that everything in the lower corner and the upper corner are considered correlated or anti-correlated. And as you can see in the bottom, you have the most anti-correlation, which we expect as DMDR as a repressor. In the upper corner, you have one binding site that is different. And that's actually expected because this gene itself, that's under the control of the EMDR, is a regulator. So the EMDR is activating another regulator to do more downstream processes. But if we dive further into these matches that are in these upper and lower corners, next slide, we get this view. We can see now from all the single targets, which are these little blue circles, the EMDR binding sites, that we made these operons. You can see from the colors that we could also link them to function. One of the things that we could see is that these genes are very often related to siderophores. Siderophores is a different name for these iron acquisition genes. So that's something that we expect, that everything that is controlled by the EMDR is related to siderophores. But then in this box, there's this one gene cluster that we actually didn't really know. We haven't seen it. It looks like it's related to siderophores, but we don't know where it's coming from. Other ones like silicillin or desferrioxamines are ones that are very well known and also predicted by anti-smash. So we're a bit surprised by this one cluster. So to go to the next slide, we looked into this cluster to also see like if our predictions work. Maybe this would be a false positive, yes or no. But from some homology searches, we could actually see that this cluster looks very similar to the desferrioxamine locus. It has similarity with genes that are connected to this same pathway. So a little bit about the desferrioxamines on the right. You have roughly two types, a cyclic and a linear type. For clarity for the next ones, I've put desferrioxamine E and desferrioxamine B there on the presentation. I will talk more about them later. And we were wondering if this new cluster that we identified through regulation actually connected to one of these variants of desferrioxamines because we see the similarity. So if you click one more time, we made knockout mutants of these three genes that show homology to siderophores to see if that actually was the case. We used CRISPR-Cas, and a postdoc in my lab was an expert on that, and she performed this experiment. If you go to the next one. Then we did LC-MS-based metabolic search, metabolic exploration of our results. We compared the well type that we see now on the upper corner to these individual genes. So if you click one more time, thank you. The first gene, we could see that there is not a huge difference between the wild type and this knockout mutant, but we could see that there seems to be a bit of an overproduction of desferrioxamine B. So that was already, like, we could see a difference. But if we then go to the next one, we could see a real clear difference in almost the complete abolishment of desferrioxamine B when we have these knockout mutants. There's a little bit of B left, especially you can see it in the lower knockout mutant for 50. So not entirely abolished, but almost completely, which makes us believe that this cluster that we identified may actually be involved in the production of desferrioxamine B, also showing that this method that we use of using regulation to discover something new worked, because this was from a model organism that people have been studying for a long time. Desferrioxamines are very well known, and still, with using regulation, we could discover something new. But for all of you, how would you apply this? Yeah, the next one, thank you. If I've made some tools, I see that some of the figures are a bit blurry, probably due to the conversion, but I try to make it easy for people to use these predictions in their own work. So it's not just me looking at my gene clusters, but it's for everybody to use. One of the things I did was making this tool called Minimotive on the left, which does all of these detection algorithms. It is a command line tool for people that are experienced with using that. They could use this tool and actually apply these methods to their own bacteria and try to find regulation for their gene clusters or their genes. I also have a web page connected to this. It's just called Locomotive, and this has a collection of all these actinobacterial regulational elements. There's other databases that have different kind of regulators, but I focus on actinobacteria because they're very complex in their regulatory system. So if you make it work for actinos, then it may work for other microbes. Too, we can move them more easily. But to make it even more easy, I also integrated this into Anti-Smash. I don't know if people are familiar with the view of Anti-Smash, but this is a print screen of the results page of Anti-Smash. So you can run your genomes. It will predict gene clusters, which you can see on the upper corner. It says overview, and in this bacteria there are 26 different BDCs being predicted. And on the slide I highlighted one SCAR4 gene cluster. And now Neil, if you click on. Thank you. You see this little pin marker in version 7, which is a binding site. So in this case, one more click. Yes, thank you. In this case, this is indeed DMDR1 from the TFBS Finder tab on the bottom, which says that, hey, this is a iron-dependent repressor, so maybe the lab, you want to activate this cluster, maybe under low iron conditions. You can even link back to the Locomotive web page for more information, but this should make it easy for everybody to use predictions. Next slide. But this is just a start. If you look on the left for this bacteria spectrumized celiac color, there are almost 1,000 different regulators, and at this point, we have only about 3%. So every blue line on this circle on the left is a regulator, and the red ones are the ones that we actually have information on. So it's not a lot. We're still working on this. So we teamed up with JGI, the Joint Genome Institute, and we're doing a large experiment across 10 different actinobacteria, looking at all these 900 over 900 regulators. And we use DAP-seq for this, which is an in vitro method very similar to ChIP-seq. And we hope with this to at least expand our information and our knowledge base from this 3% that we have to about 30%, and all this information will be publicly available in Locomotive. Next one. So to summarize, with this work, I could actually apply it to discover a novel BDC, try to really make it easy for everybody to use these tools in their own research. So it's not just me and my group working on this, but everybody could use this in their own research. And of course, we try to expand this knowledge beyond just actinobacteria by using multi-data. And if anybody has regulators that they're working on, they have knowledge on this, I'm very happy to know so we can implement that in the future. So the last slide. I would like to thank, of course, my two labs, the lab of Vasquez at the University of Leiden and the lab of Mark Smeema at Vasquez. And besides that, the Joint Genome Institute, also the people from the Denmark Technical University, which hold the Anti-Smash development team. They helped me greatly with developing the Anti-Smash implementation. So with that, thank you, and I'm open. Yeah, this is
All right. Okay. So I would encourage the next speakers to keep it within 12 minutes so that we can allow everybody to run through. And as a co-chair and house, we'll take all the questions at the end. So the next speaker is co-chair. Can you introduce the next speaker?
It's me. Hello.
Okay, it should be co-chair your meeting, I think.
The next speaker should be Ziaikin, right?
Right.
Yes.
Okay.
Do you hear me?
Yes.
Yes.
Sure. So do you see something?
Yes, yes, we see you.
I'm in my presentation.
Yes, yes, the presentation is coming up. You can go ahead, please.
Hi, my name is Zaid. Today I want to present you my project, Bittermass, predicting Bittermass from mass spectra. I'm a PhD student in the molecular recognition and chemical science lab under the supervision of Professor Mashini. One of the lab's projects was to create a database of compound that cause bitterness, BitterDB. It contains bitter compounds belonging to different chemical classes, including salts, peptides, fatty acids, polyphenols, flavonoids, terpenoids, glycosides, and many, many more. Taste plays a critical role in determining food preference. Bitter taste is one of the 6 basic taste modalities that humans can sense. It is widely believed that the evolution purpose of the bitter taste is to help animals avoid consuming toxic food. However, bitterness is not always a reliable indicator of toxicity, and there is evidence that many bitter compounds have health benefits, such as antioxidant properties and anti-cancer activity. Furthermore, alcohol bitterness can be a primary determinant through the consumption of some food. It's not always rejected. In fact, bitterness is often considered as desirable to cook in beverages such as coffee, beer, and wine. Previous research conducted in the lab indicated that over 77% of plant-derived products exhibit bitterness. However, just 95% of the molecules found in natural products have yet to be identified, and their mass spectra had not been assigned to the specific chemical structure. Our hypothesis is that these natural compounds can be classified as either bitter or non-bitter based on their mass spectra. To address this challenge, we developed Bittermass tool, a machine learning classifier that uses mass spectrum data to predict the probability of compound being bitter. Bitterness can be predicted from the mass spectrum in 2 ways. The first way is to compare the experimental mass spectrum with reference mass spectra by searching in the database to annotate the chemical structure of the compound. The prediction of bitterness is then performed using the machine learning tool BitterPredict, developed earlier in the new lab. This tool predicts the bitterness of a compound based on its structure. The second method is to predict bitterness directly from the mass spectrum, without intermediate annotation of the chemical structure of the compound. This study aimed to determine the most effective strategy. So, what is mass spectrometry? Mass spectrometry is a scientific technique used to determine the weight and identify of molecules in a sample. The process involves preparing a sample and converting it into ions, which are then sorted based on their mass-to-charge ratio inside the mass spectrometer. The resulting spectrum shows the distribution of different ions and reveals the composition and structure of molecules in the sample. This is an example of how benzamide molecules enzyme and break down into smaller substructures. The resulting ions are then detected and the plot of density versus M.Sc ratio is obtained. Each line of the graph corresponds to a particular substructure and its height indicates the number of ions detected. To obtain data for training the model, sources containing both bitter and non-bitter compounds were used. We use BitterDB database data used to train tools such as BitterPredict for predicting bitterness from compound structure, BitterEntrance for predicting bitterness intensity from compound structure, and publication reporting new bitter molecules that are agonists of TAS-14, one of the 25 functional bitter receptors found in humans. Mass spectra for the prepared compounds that were then extracted from the mass bank database, and the final set contained 3,577 mass spectra for 338 bitter compounds and 1,837 spectra for 512 non-bitter compounds. Both instrumental and big features were used to generate descriptors. The instrumental descriptors included information such as type of mass spectrum, ionization and ionization energy at which the spectrum was obtained. A beaming strategy was used to depict descriptors, dividing the entire range of n to z values into small beams of size 0.1 and recording the bits that fill into the corresponding beam. These two types of descriptors were then combined and used for model training. The training and testing process followed as a standard procedure. Firstly, the prepared set of spectra was split into training test set based on compounds, ensuring the spectra for the same compound were not separated. Next, the model was trained using trained set, followed by a cycle of 5-fold cross-validation and hyperparameter optimization to improve its performance. Finally, the model was validated on the test set. The procedure was repeated 20 times with the random separation. Based on the result obtained, all models performed similarly on the training set across all metrics. The variation in performance on the test set can be attributed by the success on the split. However, for the test set, the metrics have good values. Subsequently, the best model with the highest balance accuracy on the test set was selected. The analysis of the important features in descriptors revealed that the instrumental descriptors were the most important, despite only representing 10% of the total importance of all descriptors. The big descriptors, on the other hand, had a combined importance of 90%, which was evenly distributed across each beam, indicating that all beams were significant. Therefore, the model primarily used big descriptors. Two external sets were trained to next validate the Bittermass model. There are more difficult for model prediction. Orange indicates bitter compounds and blue indicates non-bitter compounds in each subset. The first external set was assembled based on several publications that investigated natural or other products using mass spectrometry in combination with sensory testing. The second set was based on the laboratory measurements carried out by the laboratory of our collaborator, Professor David Peterson, of Ohio State University. Based on the result, the model performance is higher for the first external set. The correct prediction of the model are shown in green and the incorrect ones in red. For some individual categories, such as vitamins or sugars, the model has low performance because they are not enough representative of the class in the training set, which will be corrected in the future. The final test for Bittermass was to examine the effectiveness of the strategy for Bittermass directly from the spectrum. According to the results obtained, the spectral Bittermass strategy was more effective than spectral structure Bittermass for all calculated metrics. In addition, the spectral Bittermass strategy is able to predict Bittermass for more compounds or their corresponding spectra than the spectral structure Bittermass strategy, as it does not have such a strict limitation in the applicability domain and does not lose efficiency in the intermediate step of compound structure annotation. This confirms that effectiveness of Bittermass in situations where the chemical structure of the compound is unknown, but mass spectra data is available. Bittermass developed and tested on the mass spectra of several national sources, and Bittermass can be continuously improved by incorporating additional data. Bittermass directly strategy can be assigned to evaluation of mass spectra of new plants. And if you're interested on our work at that, we will collaborate. Thank you for your attention, and I'm ready to answer your questions.
So is the next presenter ready? Okay. Okay, do you have the slide?
Yes, I have a slide. Let me share with you. Okay, visible for everyone?
Oh yeah, just click on it.
All right. Okay. Can I start?
Yes, sir.
Okay. Thank you. Good morning, everyone. Thank you for the invitation to share my presentation. So, without further ado, I would like to start with my presentation. So, the title is Biosynthesis Characterization and Biomedical Application of mycelial nanoparticle, functionalized with stable approach. So, this presentation will follow this outline. So, as you know, malaria is an important disease. It's caused to humans by protozoa, parasite protozoa belonging to Plasmodium. So this is a vector-borne disease which goes to transmitted to humans by Anopheles female mosquito. For sure, there's other route of transmission like blood transmission and congenital. So today, to date, we have 5 major human Plasmodium species, specifically Plasmodium falciparum is the deadliest species in the world. So this is the situation of the global burden of malaria in 2022. So as you see, in 2022, as per the World Health Organization, malaria causes 249 million cases and 608,000 deaths. Well, all this statistic was mainly due to Plasmodium falciparum, and Africa accounted for the bulk of this burden. So in this slide, we noted the graduate prediction of malaria burden from 2000 to 2020. But after that, we noted the level of this statistic in terms of morbidity and mortality. We have different causes, including the recent COVID-19 pandemics, but also other factors, including drug resistance. So the WHO recommended different drugs for treating malaria across pandemic countries. So artemisinin combination therapy is the main drugs recommended for treating uncomplicated malaria. And we have different types of ACTs. But in 2008, we noted a rising of emergence, independent emergence spread of Plasmodium falciparum parasite resistant to ACT. And recently, we noted the same scenario in Africa, some African countries, including Rwanda, Uganda, and more recently in Sudan. That's why one of the strategies to tackle malaria burden is to search and developing drugs using innovative approach. That's why nanotechnology, green nanotechnology could be a very interesting approach to achieve these objectives. So the project has different objectives. The first one was to synthesize, optimize, and characterize silver penetrant silver nanoparticle, and then evaluate the biomedical application, including hemocompatibility, antiplasmodial and herbicidal potential. So this is the plant we collected in the town of Douala in Cameroon, and we identified this plant at the National Herbarium of Cameroon. So this program summarizes the different activities we conducted for this experiment, and then we prepare plant extract using this protocol, and then we mix this plant extract with silver nitrate, to prepare silver nanoparticle, and then we optimize the synthesis of this silver nanoparticle by modifying 4 parameters, including incubation temperature, incubation time, the volume ratio of between silver nitrate and plant extract, and the concentration of silver nitrate. So after that, we use different techniques, physical and chemical techniques to evaluate the parameters of plant extract and for sure silver nanoparticles, as you can see on this slide. This is the different protocol we use for the biomedical assays, hemolysis assay, antiplasmodial assay, and larvicidal assay. So we use different software and different statistical approach to analyze the data. So first of all, this is the GCMX result of the plant extract, across extract. We identified using this technique 27 compounds, mainly 3 compounds as you can see on this slide. So after mixing silver nitrate and plant extract, we obtain nanoparticle using with sebapen pattern. We noted a rapid color change less than 10 minutes. That it might be a clue of synthesizes of nanoparticles. So the result of the optimization is the four above mentioned parameters. So based on that, this is the different optimal condition we use to synthesize, optimize the nanoparticle with sebapen pattern. And we noted the peak of absorption at wavelength 430 nanometers. That corresponds to the surface plasmon resonance. So the electron microscopy revealed this nanoparticle is synthesized as spheroidal and in shape and with a low polydispacity. So the atom content of the nanoparticles revealed most of the atom was constituted of silver and carbon. We also noted other atoms like phosphorus, calcium, and oxygen. The nanoparticle was crystalline in nature, as you can see using this ID analysis. We noted the four characteristics of the crystal at position 111, 200, 220, and 311. And the Fourier infrared transform spectroscopy revealed different peaks corresponding to many functional group like OH stretch, CC stretch, CC bands. This type of compound are characteristic of functional group, are characteristic of different phytocompound like polyphenol, aromatic compound, and polysaccharides. The zeta potential and DLS analysis revealed the nanoparticle was very stable because as you can see, the value of the zeta potential was stored in the reference value minus 30 to 30 millivolts. Regarding the chemotoxic activity, when we analyze the chemotoxic activity after 30 minutes and 34 hours, we noted two main conclusions. The hemolysis rate is dose and time dependent for consistent, including nanoparticles. Chloroquine, which is the standard drug, and plant extract. And the nanoparticles has the hemolysis concentration at 50% comprised between 250 and 500 micrograms per unit. That means this nanoparticle we synthesize to be very hemocompatible. When we analyze the plasmodial activity of this nanoparticle, with nanoparticle against the two plasmodium fasciclone strain, the sensitive strain 3D7 and the resistant strain RTL9. And we know that the nanoparticle was more effective than the plant extract. The nanoparticle was highly effective because we found IC50 less than 20 micrograms per ml. And the nanoparticle was more effective on 3D7 strain as compared to ECAR9. So the larvicidal toxicity revealed against these three different mosquito species is responsible for the transmission of different pathogens, responsible for different diseases like chikungunya, like malaria, like yes, chikungunya, malaria, and so on. So we noted the potency of the nanoparticles increase with the time. We follow these activities over 3 days, from 24 hours to 72 hours. We noted the nanoparticle was more effective than the plant extract. As you can see, the XD50 is less than 20 micrograms per ml, as found for the Plasmodium parasite. And this is some of the main morphological deformities we found when we tested the nanoparticle on the mosquitoes. We have different activities like the pigmentation of the body. You can see the this dark body as compared to the reference with the nanoparticle-free milieu. We can conclude based on this study, the synthesis of nanoparticle was rapid, and the nanoparticle was polycrystalline, mostly spherical, well dispersed, stable with the role of plant compounds such as alcohol, amide as reducing on trapping agent. So the nanoparticle was highly potent against Plasmodium falciparum with a low toxicity against red blood cells, thereby outlining a good hemocompatibility. And finally, the nanoparticle was also highly potent against mosquito. So I cannot finish this presentation without thanking all the funding agencies and the institution who take part of this project during my stay in India. So I also want to thank the supervisor of this project, Dr. Vineta Singh, Professor Vina Pandey, and Dr. Ravindra Kamaraju, who led the parasite and vector experiment, and also my collaborators, Dr. Fidel Ivanakidi, and Mr. Joseph Awada, and Dr. Deshabide. Thank you, and open for the question.
Thank you very much, Dr. Fosso. That was brief. You had a lot of stuff, and you gave the material in short time. Can we take the next presenter? Wilberforce. Are you ready?
Yes, I'm ready.
That's great.
Good afternoon, this afternoon. I'm Ndarawait Wilberforce, a master's student in University of Embu, Kenya. Here to present part of the research we are doing, working on in-silico search for alpha amylase inhibitors for the management of postprandial hyperglycemia. So diabetes is one of the major metabolic illness. It is normally characterized by unbalanced sugar levels in the body. The disease has been characterized in two, normally as type 1 diabetes mellitus, diabetes mellitus. But World Health Organization in 2016 confirmed that among the patients that were found having the disease, 90 to 95% of them were as a result of type 2 diabetes. And the mortality globally in 2017 is about 7 million people, as per the research done by the site. And looking at this, in terms of the diabetes, it's a result of being as a result of the blood sugar level that results from the digestion carbohydrates in the body. And in that case, there's normally some enzyme. Before that, so the enzymes that are involved in digestion, including the alpha amylase, we have alpha amylase and other associated enzymes that are involved in breakdown of the complex polysaccharides by the simple sugars that absorb and assimilated in the body. The regulation of these sugar levels, regulation of this enzyme help us to regulate the ability to digest and make available glucose that could be absorbed. And now, in an healthy human being, we normally have the homeostatic function of the body regulating the sugar levels using the insulin and the glucagon hormone depending on the fluctuation. Like this. So we normally have the drugs, the available drugs already that could be used to control the digestion of these enzymes, that they work through inhibition. We have the acarbose, we have the miglyton, we have the glucose, and even the metmorphin. But most of these drugs have been associated with a lot of side effects, including the stomach upset as well as the effects on the liver, and others even could lead to hypoglycemia, taking too low the sugar levels. And therefore, this could lead to pressing need to come up with more alternative inhibitors. But coming with more alternative inhibitors through the experimentation models, experimental laboratory experimentation would be a bit challenging as it takes a lot of time and it's quite expensive as it is involving trial and error to come up with a drug. And therefore, in our research, this will get us to try to leverage on the databases, as we have most of the database having metabolites are already in the online. We have the zinc 15, we have the drug bank, we have the PubChem, as well as the past database, the productive species database, as well as the NMR. So we could leverage on this database and we put the virtual script so that we have an alternative. The method we adopted is that we picked one database, that is natural product activity and species database, and then we selected a receptor. In this case, we picked the alpha amylase as one of the enzymes that is involved in digestion. Then we acquired the enzyme from protein data bank. Then we took the database and we prepared it to the curation so that we can remove the molecules having bad geometries, as we downloaded all the database having 10,000. So we prepared the bad valencies and the duplicate molecules, as well as converting the database from to 3D conversion to 3D database using the OpenBabel. And then now we did the virtual screening through the drug-like and kinetic properties that is using the Metchem mnemonics that include the basic rule of 5. We have the Egan rule, Booth rule, rule, as well as the Mekan rule, so that we trickle down the 1,906 molecules. Back all the molecules that could act as the lead type molecules will remain with around 2,250 molecules. Then we subjected the toxicity screening using two servers. We used the data warrior as well as the CAiSMD server, that we were able to filter out all the molecules that so the endpoints based on those one like the MS toxicity, mutagenic, tumorigenic, and so forth. Then the remaining 576 molecules we subjected to molecular docking using molecular operating environment. So upon doing the virtual screening of the pharmacokinetic properties, we are able to have more than 2,000 molecules and their properties as per distributed based on the factors we considered in the pharmacokinetics, including the hydrogen bond donors, number of rotatable bonds, hydrogen bond acceptors, molecular weight, all those we considered, and then we subjected to origin so that we can have the graphs of the remaining molecules to see the distribution of the properties. Then in terms of the toxicological properties, we first subjected the remaining molecules into data warrior as well as the CAiSMD. And any molecule that will have the potential of being toxic depending on the endpoint like skin sensitization, toxicity, HER3 inhibitor as well as M toxicity, and even on the data warrior where we are looking at mutagenic molecules that cause cancer, superigenic reproductive effect as well as irritants. So any molecule that show either being low or high were knocked out as being toxic so that we remain in 507 molecules we subjected it for docking. Then before docking, we had to download the receptor from the protein data bank and we prepared it in terms of adding the missing nitrogen, that is as well as the side chain, building any missing groups like that could have more than 10 residues in them. Then we protonated so that we can optimize the ionization state of the molecule. But first we had to tether using the mode so that we can retain the original geometry of these atoms as captured by the crystallographic structure. Then any atom fall outside the active site fix of the residue composition efficiency. Then we did the energy minimization in terms of the refinement. But before that, we identified the active site, and in this case, the active site, because the molecule we picked was having a carbon at the co-crystallized. So we picked our active site being the where the co-crystallized carbon was, so that we can dock our molecules, the remaining molecules inside the active site where the co-crystallized. But first, we validated the docking by redocking the carbons inside the active site, and we tried to overlay the docked one and the co-crystallizer to see the one that can overlay and have the exact position where the co-crystallizer will be, so that we can validate and see our molecules being docked in the active site. For docking, we filtered out molecules based on the results of the validation docking. That was negative 12.55 kilocals per mole. So we were able to see two molecules, that is NPC44580 and NPC1317, having higher binding affinity more than that of their carbons. And then even the root mean square deviation was below 2 recommended. And looking at in terms of the interaction, we realized that these molecules could be able to interact with most of the amino acids, including the aspartame 300, glutamic acid, lysine, aspartame is, and NPC 1317 were able to interact having the hydrogen donor, what kind of the interaction with aspartame 300, aspartame 177, and is 299 as well as adenine 195. So looking at these amino acids, you realize that most of them, all of them were those that were in the active site of the receptor. And now we have generated the 2D interaction as well as the 3D confirmation of the ligand as well as the receptor, the way it looks in the active site. And here we can see the graph, the 2D interaction having the site receptors, the properties depending on the amino acid, the polar acidic, the seq of the residues that interacted with the ligand, and even the confirmation of the ligand to the active site of the NPC44580 Right? So this is also the other interaction of the other ligand, the active site, with the receptor also. Then in terms of conclusion, so we realized that we were able, leveraging on the past data, we were able to identify 2 ligands that could act as a potential drug bikes, and the modalities we used for virtual screening, we adopted the drug bike, the delay assessment using the mnemonic beyond rule of 5 that could encompass several kinds of mnemonic. That is a can rule, a goes rule, a Bensky rule, perhaps a Megan rule. And because of that, we're able to identify 2 molecules as promising as diabetic agents. Next slide. What we propose for the future studies is additional experimentation in terms of the in vitro and in vivo studies to ascertain the diabetic efficacy of the 2 identified molecules. Acknowledgement. I cannot take granted to say thank you to my supervisors, Dr. Jude from University of Embu, who is currently in Helmholtz Center of Infection Research in Germany, and Charles Otieno from Maseno University in Kenya, and Dr. David Kitheka from Metabolite University in Kenya. Thank you so much.
Wilberforce for good time management and for the brief talk. Can we get Dr. Donatus? Are you ready?
Yes, I'm. Let me get my slide.
Does every presenter know the sequence where you belong? So that you get ready. Okay, thank you, sir.
Yeah, thank you so much.
Right.
Thank you so much. The work I have the honor to present is part of our ongoing work. Very soon we are going to have it in the press. It has been accepted already for publication. The work is titled Design, Synthesis, Biological and Computational Screening of Novel Oxindole Derivatives as inhibitors of YI kinase and SARS-CoV-2 spike post ACA2 interaction. We are going to have this outline for the presentation. Cancer is a life-threatening disease that has killed millions of people each year passing by, and in spite of several efforts, it has continued to resist full control and eradication. But interestingly, YI kinase is multifunctional proteins highly implicated in cancer, and it is important in mitotic progression. And if this protein is interrupted, mitotic progression gets interrupted. And this may lead to the death of malignant cells. And during the outbreak of COVID-19, there was widespread interest in the repurposing of most anti-cancer drugs as potential inhibitors of severe acute respiratory syndrome coronavirus 2 spike and host angiotensin-converting enzyme 2 interaction as a strategy to prevent these viral enzymes. Considering that ACA2 also serves as a receptor, the receptor for the SARS-CoV or SARS coronavirus 2, the binding at this S1 domain of SARS coronavirus spike protein to ACA2 initiate viral entry. So this area is very, very important for therapeutic interest in developing possible treatment for COVID-19. Why this oxidized indole? Isatin is a secondary metabolite of tryptophan, and can be found in a broad range of natural and synthetically derived pharmacologically active compounds. And it has been proven to have antiviral and anti-cancer properties. This cystatin moiety can be modified at the N1 position, the C3, the C4, the C5, 6, and C7 position. But modification at the N1 and C3, N1, C3, and C5 positions are much more favorable for diverse biological activity. So the main aim of this work was to design and synthesize this novel hybrid of cystatin and then evaluate their biological activities as inhibitors of YI02 spike host ACL2 interaction, with specific objective tests to design, synthesize, and characterize these analogs of cystatin. Then, carry on the biological evaluation of the compounds as inhibitors of these two targets and mechanism of action of these hybrids using computer-aided methods. For the design of the compounds, focus was more at the C3 and the C5 positions, resulting to the amide biocompounds and the amide hydrazone compound. The synthesis of the compounds follow the synthetic scheme. And at the third step, the first target compound, that the amide biocompounds, were achieved. And the last, we have the second target, the amide hydrazone compounds. These compounds were then characterized using LCMS, both proton and carbon 13 NMR, to elucidate the structure, to establish the structure of the compounds. And summarily, we had 6 amide spiral compounds and 12 amide hydrazone compounds. Compounds were then submitted for biology based on the 2 targets. The disruption of the activities of YI kinase was established using a homogeneous time resolved fluorescence kinase assay, which can measure YI kinase 1 activity by phosphorylation. And the result obtained were able to show that the control espiradine would inhibit YI kinase with an IC50 value of 5.3 nanomole. And this was consistent with previous reports. Several of the compounds were able to inhibit YI kinase with DBE01, 031, 039, and 059 having very interesting activity. The compounds were also tested to disrupt the interaction of SARS-CoV-2 by protein, that is the original one virus, with the host, angiotensin converting enzyme 2 receptor using alpha screening. Here, 2 recombinant proteins, that is the spike receptor domain and AC2 protein, were used, and each fused to a donor and a receptor bit. When the binding between the protein occurs, a singlet oxygen transfer occurs between the bits, which promote luminescence. Compounds that can inhibit this interaction also inhibit luminescence. And the control that was used for papinol would block this interaction with an IC50 value of 0.3 micromolar, which was also consistent with published values. Only 2 of the compounds were able to show interesting anti-SARS-CoV activity. were able to, with IC50 values, comparatively lesser as to that of the control. Now, to establish a mechanism of action, we first establish a protocol by docking validation. By docking validation, which consists in redocking the native or the co-crystallized ligand for each target protein. This was necessary to ensure that the docking produce procedure is able to reproduce the binding mode of the inhibitor for both the target proteins. And this docking validation, interestingly, gave us a docking a root mean square deviation of 0.5 Armstrong, which was very, very encouraging. And we therefore adopted this established docking protocol. The superposition of the docking of the active and inactive compounds for both targets, that is the YI kinase, and activities and the spike ACA2 interaction, so that the active compounds in deep purple for both cases and the inactive ones in light orange bind differently overall. The orientations of that of the deep purple and the light orange were completely different. And looking very closely at the different interaction for the active compounds, for the case of YI kinase, it shows that both interact with leucine 139 and alanine 213. And it has been proven that YI kinase domain consists of a hydrophobic pocket composed of leucine 263, glycine 216, and leucine 139. Besides this, the amino acids of that is the alanine 213, 137, are known to assist in anchoring the ligand to direct them in its hydrophobic pocket to a hydrogen bond formation. It was also shown that the small molecules which the hydrophobic features towards the hydrophobic pocket of the receptor are crucial for YI kinase inhibitory activities. For leucine 139, this residue interacts with the amide functional group of the active ligands. And for alanine 213, it's seen to interact with the core of the insertin moiety. For all the compounds that were active for YI kinase. Meanwhile, it was observed that all the active compounds make interactions with lysine 162 and glutamine 211. And these two amino acid residues are very important for activity. For the computational study performed on the Y type or the Wuhan variant of SARS-CoV-2, the docking study showed that the ligand preferably binds to the ACA2 receptor binding site, hence preventing the recognition of the receptor binding domain of the viral spike by the host protein. Only two of the tested compounds showed activities are against the spike, ACA2 fusion, and even show weaker activities when compared to that of the inhibitory concentration against YI kinase. And this could be explained relative to the size, in the small size and weaker hydrophobicity of this ligand when compared to the much bulkier cochlear ligands in the case of the reference control phosphophenol. Yeah, this work, we have several analogs of certain that were synthesized and characterized, and we established the inhibitory activity for both YI kinase and SARS-CoV-2 by post-ACA2 interaction. Among the synthesized compounds, several of them exhibited YI kinase inhibitory activity, whereas only 2 of these compounds were able to block SARS-CoV-2 spike ACA2 fusion. This work meant to prove the concept that most existing cancer drugs possess antiviral properties. And the molecular model showed that they have for each protein adopted different binding modes, hence interacting with a different set of amino acid residue, the binding site. The weaker activities against the spike AC2 to be explained by the very small sizes of these compounds that fails to address the important interactions for binding to the ACA2 receptor site. And for perspective, we plan to increase the chemical space of these compounds by starting with building the pseudo of this cysteine and why not indazole hybrids as potential anti-cancer and antiviral drug agents. I want to extend my thanks to my university, University of Oya, of course, the Center for Drug Discovery, the Winster Institute, where this work was carried out, and we acknowledge funding of this work by the Bill and Melinda Gates Foundation. Thank you so much for your keen attention.
Thank you very much, Dr. Donatus Eni. We plead that the next presenters will try to be time conscious. We want to find out whether Bruno Akamba is ready. Bruno Akamba, are you ready? Is he there? All right. It looks like Bruno Akamba is not there. The next is Obin Yubi Kamche. Is the presentation ready?
I can hear Bruno speaking.
Oh,
Bruno was speaking already.
oh, that's great. Then why can't we hear you?
Can you talk louder?
Is that a slide which is showing
Yes,
on the screen?
This is Bruno's slides.
Okay, that's fine.
Can you start, Bruno, please?
Oh. His microphone may be having a problem.
It looks like it's a recorded. It looks like it's a recorded. It's a recorded lecture.
Is it there?
Yeah.
Do we go to the next?
Yes, we can move ahead.
Marshall Kyle Ayeno. Ayeno.
I don't seem to see him connected. Bruno, are you there? Bruno, I see Bruno connected, but he sent the slides that are already with the lecture already registered in it.
Oh no, then we can keep it at the end. Yeah. So let's take Mr. Beto too. I guess it's ready.
Jude, if you're there, please go ahead.
Good morning, everyone. Can you hear me?
Yeah, we hear you. We cannot see your slides.
Okay, I'm sharing my slide right away.
Prof, you have to stop sharing that slide.
Okay, this is what I want. Yeah, I stopped. Okay, I have to stop sharing it. So is it fine now? Is it fine now with you? Good.
Is that Jude, Beto?
Yes.
Okay, go ahead.
Let's can you move to presentation mode? Just click on slide share and move on, please.
Okay, can you see it largely now?
Not yet. It would be best if I get all the slides in because it's a lot more efficient.
Yeah, you're right.
Just click on slide share and we'll start working. Otherwise, you send me your slides and we'll start. We can save time.
Yes. Find out.
I thought
it is not. You go to the PowerPoint slide itself and click on slide share and it works. Please, the next person the next speaker should send their slides. When you don't send your slide ahead of time, it's quite difficult for us to manage this because it has to do with time.
Yeah. So we expect David Kitheka.
That was the assignment. That was the announcement made in the morning. I will send several.
O.Obende and Daniel Moscow Ajere Torah to send us later.
I have sent
Those with us in-house. I will be presenting on the design and implementation of a unified version of the African Natural Product Database. Some of us have been carried out a hands-on session on this database yesterday and talk more about it today. So this is a histogram of the previous developments of natural product libraries from Africa. Some of these databases are much more specific. You have Afro-Malaria TB that contains a database of malaria compounds. You have CAMMED and COMMED that are natural products of Africa. And what is found in ANPDB, you have the non-PTB, and you also have EANPDB, that is natural products from East Africa. Currently in ANPDB, we have 6,515 compounds that are found in the database, and we found it necessary to update the database with new entries. And in these past few years, we have collected literature data from various sources. Most are journal articles, and Some of them are manuscripts from Ph.D. and M.Sc thesis. So while collecting the data, we collect information such as the name of the compound, the source from which the compound was related, the date at which the compound, where the compound or where the source is available, the SMILES of the compound, the ID, and also the compound class and the subclasses. For example, if you have phenols, you know the various subclasses of phenols. You have diterpenes, triterpenes, for alkaloids, you have indole alkaloids, urano alkaloids, and so on and so forth. So those are the type of information we are recording. So later we will have to get the pharmacokinetic profiles of these compounds and as well as toxicity profiles in order to have our updated ANPDB. So in the in-house data, we have information about the data that we have collected, and we have arranged them in regions, Northern Africa to Western Africa. And we can see that from our data collected, we have 6,006 compounds which are unique. And concerning the diversity of our database, we have in Central Africa, in the Central African region, we have 24% of our compounds that are terpenoids. We also have in Northern Africa 26.4 are also terpenoids, and Western Africa 40% of our data are alkaloids, and in Southern Africa is 41.1% are flavonoids. Also in East Africa, 32.1% make up the highest percentage of our data in this region. So this shows how our data is quite diverse, and it is quite a good news when you want to synthesizing a variety of compounds for various tasks. So we also collected small strings of our data. But before doing that, we have to double check our entries to make sure that no errors were done while doing the curation. And in collecting the SMILES, some of our compounds are present in PubChem, so we also have to note down the PubChem IDs. And from the PubChem IDs, we make use of Python scripts and OpenBabel in order to obtain their SDL files and then extract now their SMILES from there. And for compounds that don't have PubChem IDs, we'll have to draw them using ChemDraw, but you can use other softwares to draw these molecules like Marvin Sketch and so on. So from the MOL files, we could also convert their structures to obtain their SMILES, and from there, we now create some Excel files or CSV files that will merge now in order to calculate now the physicochemical properties and the toxicity profiles of our compounds. So this research is ongoing, and what we'll do next is use the appropriate tools in order to get their physicochemical properties and the toxicity properties, and also we'll have to prepare our data well and upload using SQL. So by the time we unify our data, we'll have about 15,000 compounds in the updated version of our database. So in conclusion, this database could be applied in the diverse area, molecular logging, fragment-based drug design, can also be used to investigate the chemical space of your data, depending on the biological target you're trying to investigate. You can see how your compounds are could be used in order to investigate the activity against that target. We could also use this database to build bioactivity predictor models based on the activity of choice and the SARS-CoV and HIV, other bioactivity. It could also be used for the de novo design of pseudo-natural products or natural product-like compounds. Because with natural products, at times we isolate these compounds, we mostly get very low yields, and their structures are quite complex to synthesize. So you can use the de novo design in order to get natural product-like compounds, and why not have synthetically accessible compounds. And also, these proposed compounds will also be synthesized in order to enhance the activity compared to the parent natural. So this research would have not been possible by the funding of the Bill and Melinda Gates through the Carrington Fellowship that was offered to our PI and my supervisor, Professor Fidelity. I would love to thank also the University of Goya, the postgraduate students, and our partners, some of our partners, Celia and the Research Institute, who were here last year to train us on AI models and also bioassay technology, and also for the TMS Foundation for giving me the opportunity to present this work. Thank you very much.
Thank you, Mr. Beto. That was brief, and thank you for time management. We'll skip some presenters like Mr. Bruno Agamba. Are you ready now? Thank you, Yobi. Is he there?
Yes, I'm there.
Okay, all right. Then we can have you. So you can start. Obi, you should unmute yourself.
Yes, please, can you see my slide?
Yes.
Unfortunately, we cannot use the slides because they have video recorded in it and it's causing voice to overlap. It's better that he sends us slides that we don't have. Okay, if it's rich, that's fine. So you can go ahead, please, from there, because from here there's voice in overlapping. So you can present since his slides already loaded.
Okay, thank you for giving me the floor. The work I have the honor to present to you this morning is entitled Targeting the Intra-Erythrocytic Life Cycle, the Malaria Parasite, Identify Starting Points for Anti-Malaria Drug Discovery from Rimaya Horata and Makaranga Munanga. It will be presented to you according to this plan: introduction, material method, result, conclusion, and outlook. As you already know, malaria is an infectious disease transmitted to humans mainly by the bite of a female mosquito called Amylase. Among the species that are responsible for the disease, Kojom falciparum is the most dangerous. And a study of its life cycle showed that when the mosquito inoculates sporozoid, they infect hepatocyte where they develop into schizome. and the rupture of carbon liberate new merozoid which infect erythrocyte, where they develop asexually into three main stages: grain, trophozoite, and schizoid. And the rupture of schizoid liberate again new merozoid infect another erythrocyte for the intra-erythrocyte life cycle to continue. And knowing that this intra-erythrocyte life cycle is responsible for the clinical manifestation of the disease, such as headache, vomiting, muscular pains, weakness, diarrhea, fever, and so on, targeting this intra-erythrocyte life cycle is promising in the searching for new anti-malaria drugs. The management of this is mainly relied on artemisinin-based combination therapy. However, these drugs face many constraints, such as the cost and accessibility in rural areas, the side effects, and mainly the development of resistance phenomena, highlighting the necessity to find for new drugs against malaria. In this respect, natural product can be a promising source of compound for modern drug discovery. We observe that Primaya podata and Makaranga monanga are two plants traditionally used in Cameroon to treat various ailments, including malaria and its related symptoms. And to the best of our knowledge, no previous studies have been carried out on the search for molecule against Plasmodium falciparum using this plant. We therefore hypothesize that Primaya podata and Makaranga monanga extra are a source for the development of new anti-malaria drugs. The general objective of this work was therefore to target the intra-erythrocyte life cycle of the malaria parasite to discover new drugs from Primaya podata and Makaranga monanga. Specifically, we have access to
Mr. Gobin, your slides are not moving. We are still on the title slide. And
maybe this, let me fix it.
Yeah.
I tried to send it to Professor Fidel, but the size was very, very high and it could not be shared by email.
I got the version is sent. I got the version is sent, but unfortunately, it's recorded with video in it, with audio in it. It's audio containing slides. So when I upload it, I hear your voice talking, and then also you are also presenting. So there's conflict of voices, so I could not upload it. Otherwise, we give you some time to fix it up, and we call the next speaker, and we can get back to you. Since I have the
okay, okay. Is it possible for you to use the slide that he will be talking?
Yes, but that is the idea. But the slide is sent to me has audio recording in it.
Okay,
yeah, that's the problem. So, which means that there are two audios talking at the same time. Otherwise, if you don't mind, we can call for the next speaker, and while before then he would have fixed this issue. David Kitheka, I got your slides here. Kitheka.
Okay. Is it ready?
Yes, it's ready.
Yes, I'm ready.
All right, you go ahead.
Hello, everyone. I'm Kitheka David from University of Embu, Kenya. I'm going to talk about chemistry and the pharmacoinformatics of compounds related from Vernonia compounds. Next slide, please. My plan of presentation, I will briefly introduce my topic, methodology, discuss the results, and then conclude. Next slide. Next slide, please. Next slide, please.
In the slides.
Slide number 3, please.
I think we are there already. Want me to go further? That's the next one.
From my side, I'm in slide number 5.
So 2, 3. Is that fine?
No, I'm still in slide number 5.
Oh no, something's wrong.
Yeah.
Because if you are number 3, this is number 3 on my screen.
Maybe something be delayed.
Oh yeah, that means that it's an internet forum because on my side it's a number 3.
Okay.
Yeah.
Now I'm on number 3.
Yeah, fine.
Grants can be sourced from various sources, such as natural sources, where we can obtain grants from plants, microorganisms, and animals. Similarly, we can obtain grants from through synthesis and through doing some chemical modification of natural product to increase therapeutic properties. Among these sources, natural products have been widely explored and used to treat various infections. And according to Najim and others, approximately 35% of commercially available drugs came from natural products. This made natural product subject of interest for researchers. As a result, the database for natural product has been extensively increasing, with millions of compounds having unknown activities. Similarly, in recent years, there has been decline in interest of natural product. This is because of challenges associated with isolation, characterization, low yield, and generally the process is time consuming and expensive. Then the big question is, can artificial intelligence revolutionize natural product research? Next slide. Computational, in recent years, there has been a surge of computational application in drug discovery. This process involves a virtual screening of large chemical libraries to identify potential drug compounds, optimizing each of the compounds, evaluating the toxicity, and lastly, validating this compound through in vitro and in vivo experiments. However, despite this being known, even from what we have witnessed in earlier presenters, pharmacokinetics and animal profile have been briefly investigated. Therefore, we evaluated the chemistry and pharmacoinformatics of compound derived from Vernonia species. This is because Vernonia species have been traditionally used to treat in various purposes, such as traditional medicines, and some have been investigated for their biological activities and have shown potential pharmacological properties. Next slide. To achieve this study, we collected 677 compounds from literature, and we grouped them according to their respective subclasses. Then we generated their chemical structures using Keynote. After that, we copied this generated compound as mass into Satini, which predicted drug likeness properties. Similarly, we copied the generated structure as mass into CAiSMD website, which predicted their toxicity. Then this data was retrieved into an Excel spreadsheet where it was statistically analyzed. Next slide, please. Next slide, please. Next slide, please.
Are you okay with this?
I'm still in methodology.
Yeah?
I'm still in methodology. Next slide.
This is a discussion, right?
I'm in slide number 5.
Yeah, because there's a problem of internet connection between me and you. Actually, I'm on 6 p.m. So apparently you're not getting me.
So they can go a slide behind.
Okay, I'm now on 6, right? Is that what you want?
No, you
Yes,
want
yes.
methodology.
I'm okay now.
Okay, fine.
Yes, this study field revealed that genus Vernonia contains various phytochemical compounds, such as with terpenoids, flavonoids, and alkaloids being the most prevalent. We went ahead and did some analysis to identify the most common basic structures. The results revealed that structure scaffold number 1 and number 2 are the most promising in this genus, where we identified scaffold number 1 is for sesquiterpene lactones, while number 2 is for flavonoids. Similarly, it was reported that these compounds contain various bioactivities, such as malaria, anti-inflammatory, antioxidant, and anti-cancer. However, anti-inflammatory and antibacterial are the most prevalent, are the most observed activities. We went ahead to analyze the drug likeness properties, and it was observed that most of these compounds have properties that are within specified limit. And we observed that, next slide. We observed that 92% of these compounds absorb the basic rule of 5, while 67.4% obey Campbell's law, while 58.8% obeyed Ingham's value. We went ahead and did absorption prediction, where we analyzed being glycoprotein substrate. The results revealed that 51% of the compounds obey end-low glycoprotein substrate, which means that most of these compounds can actively transfer from the cell by glycoprotein to blood streams. We did metabolism prediction, where we analyzed inhibition of cytochromes, and as shown in this bar graph, most of the compounds end low inhibition of this cytochromes. Lastly, we analyzed the data for toxicity level, and most of the compound end low toxicity level. Next slide. Next slide, please. Next slide.
I am pretty sure that I'm in the right slide, but your internet, they are quite not on the same page. If I now?
Yes, fine now. We went ahead and we compared the drug likeness properties of these compounds with those of approved FDN drugs. We found that most of these compounds cluster closely together those of approved FDN drugs. This suggests that these compounds share the same characteristics and they have potential for clinical success. Lastly, we went ahead and predicted their synthetic accessibility to evaluate the feasibility of synthesis based on their structures. We found that flavonoids, sesquiterpene lactones, triterpenoids, and monoterpenoids are difficult to synthesize as compared to other compounds. Next slide. In conclusion, phytochemicals of genus Vernonia exhibited physiochemical profile comparable to those of FDA-approved drugs. This highlights their potential suitability for further investigation as promising drug development candidates. This generally underscores the value of computational method in accelerating drug discovery. Thank you for your attention.
Thank you, David. Can we still verify if Marshall Payeno is there? All right. O.Obende.
All right, so I share O.Obende slides. Okay.
Sam Felix O.Obende, are you ready?
Yes, I'm ready.
That's great.
Hello? Yes.
I'm now the one having technical problems. Okay. All right, let me make it larger. Okay. So, good.
We can see it.
Yeah, just give me a moment. I'll be there. Cool. It should work now.
That's great.
Yeah, can I start?
Yes, sir.
Good afternoon, everyone. Here in Kenya, it is afternoon. So, I'm Sam Felix O.Obende, master student from Maseno University. I'm going to present work that is part of my project that has been done. That is talking about in-silico analysis of ADME tox profile of compounds from croton species. Next. So under this species, we have around 1,300 species worldwide that has distributed. Move on. And as you can see, in figure 1, there is reports showing the articles that have been reported on the croton compounds. And the articles are compound isolation and activity. As you can see, the blue color, while the orange one shows compound isolation only. So this graph helped us to come up with the time frame on the research. As you can see, between 1990 to 1999, there was an upsurge increase in articles that covered compound isolation and activity, and at the same time compound isolation only. So that is why our research covered mainly from 1990 to date. As you can see, from 2020 and beyond, it is reducing down because the time frame is very small. The time frame was 10 years, but because here it is 2020 to 2024, around 4 years, not so much articles have been reported about croton compounds. So that gives us the smaller comparison. As you can see in B, it is the ethnomedicinal uses of these croton compounds. And as you can see, the majority being the gastrointestinal, which is having around 39, while the I1 is anti-inflammatory as reported according to the count. So this one tells us that these compounds are very, very good because they were used traditionally for these different disorders, diseases. Next. So because there has been ethnomedicinal uses and biological activities, there has not been ADME properties evaluation of the compounds isolated from croton. And this work emphasizes on it focused on compiling latest findings from the literature on the chemical components of croton species that were obtained from Google Scholar, Scopus, Science Direct, Biomed Central, and PubMed. So it was a thorough search. All the compounds that have already been reported from 1990 to date were searched and then accumulated to come up with the metabolites. And also, we obtained Food and Drug Administration from enamine.net.com. to obtain our FDA drugs. Next. So after that, then we use Swiss ADME and PKCSE to obtain ADME toxic properties, whereby ADME properties were obtained by using the Swiss ADME and toxicity properties were obtained using PKCSN. After that, there was statistical analysis using origin probe to come up with conclusion. Next. So, what are the results? These are the metabolites present in the croton compound from 1990 up to now. And as you can see, the majority being the terpenoids, followed by alkaloids and others. And under terpenoids, 67.2% were diterpenoids, and then we have monoterpenoids, sexterpenoids, and other terpenoids. Next. So graph B showed the representation of the biological activities. This is just an estimated, it's not it is an estimated, those that have been reported, though I did not get the actual one, but this is just an estimation of those that I got from the literature. And it is sum up to 100%. So as you can see, out of those that have been estimated, majority Y cytotoxic, followed by anti-inflammatory and others, as you can see. Then after getting the pharmacokinetic properties, we used it in the origin pro to come up with principal component analysis, whereby we obtained the principal component analysis 1. This is the major variance, major relation according to the variance, and PC2 is the second variance, those that are not captured with PCA1. So it shows that you can see this red color shows the croton compounds, while the FDA were the black dots. As you can see, majority of the croton compounds lie in between the FDA part. So it shows that there's a relationship between the croton compounds and the already FDA drugs that are available in the app. So after that, there was drug likeness and lead-like assessment by use of ITMAP. The ITMAP was we generated was having rule, whereby rule we had around 6 rules: the Lipinski rule, the Gauss rule, the Weber's rule, the Egan rule, the Muege rule, and the lead-like rule. Well, the lead-like rule is the rule beyond, is the one beyond rule of 3, rule of 5. That is the rule of 3. And the Lipinski rule mainly covers the parameters, the physical parameters of molecular weight less than 500, and the Egan rule just covers the log P, that is the lipophilicity, that is will be less than 5.58. So as you can see, the red color shows that 0 violation, which means proton compounds that were not violated. They are good for drug like, which means they were good according to molecular weight. They're also good according to lipophilicity. They're also good according to hydrogen bond donors. While at 5, that is the blue, shows 5 violations. It could violate lipophilicity, log P lipophilicity, could violate the molecular weight, which means molecular weight greater than 500, and it also could violate the hydrogen bond donors, rotatable bond of a compound. So this tells us that although we have these compounds, but some can be repeated. We can have one rule having 2 being violated, while the other rule being repeated having 3 or more 5, as you can see here. Next slide. So from drug-like properties of the heatmap, we came up to obtain other ADME properties like bioavailability, PG substrate, gastrointestinal absorption, blood-brain barrier permeability, and pain. As you can see in the pie chart 1, it shows the bioavailability. This bioavailability shows that the majority of the compound can be easily absorbed. And bioavailability is the ability of the compound to reach the target site in its active form. So as you can see, 88.5% are good compounds, and they reach in the active part in the body in active form as opposed to the 0.11, which is black, they are 6.2%. While in the graph B, that is in graph B, we have the pain, BB permeability, PG substrate, gastrointestinal absorption, and it is in a binary form. We have yes and no. As you can see, the BBB permeability is those compounds that can penetrate through the brain. They can be used as good drugs. Thus, they can really penetrate to the brain so that they can be taken to the right part for them to act as drugs. So, as you can see, majority of the compounds, around 56.62% are good BBB permeability. 42.81 are good PG substrate, while 83.56 are good gastrointestinal absorption, and only 3.42% of the compounds were acting on pain, according to the anesthetic of pain. And then under the enzyme inhibition of graph C, majority of the compounds were not inhibited. So, which means they are good to be used as drugs. Next. So, from the ADME, then we go to the toxicity prediction. We predicted how these compounds were toxic according to 5 endpoints. That is the Amylase toxicity, HEG1 inhibitor and 2, hepatotoxicity and skin sensitivity. We predicted them and come up with a conclusion that the HEG1 inhibitor, all of the compounds were not toxic according to that, while few of the compounds were toxic, around 25.3%, 34% were toxic against HEG2 inhibitor. And 10.09 compounds were toxic according to Amylase toxicity, and skin sensitization, only 90.15 compounds were good. They were not against skin sensitized. In conclusion, the major class of compounds from croton are diterpenoids, and the main biological activities reported are cytotoxicity, that is 52%, and anti-inflammatory activities, which is at 13.4%. According to the drug-like rule, 76% of the compounds were drug-like, which means they obeyed the rule of 5. And croton compound share a common chemical space as per the approved FDA drug. That is according to the PCA analysis. And last but not least, ADME analysis proved only 39 compounds to be viable drug-like. 39 compounds to be drug-like out of the compounds that we had. And when we did toxic prediction, 20 compounds were knocked out, which means 19 compounds were non-toxic. Recommendation, further docking and simulation should be done on the 19 compounds noted for their respective biological activities reported. The last one, I want to acknowledge my supervisor, Dr. Charles Ochieng of Maseno University, Dr. Emmanuel Shikanga of Maseno University, Dr. Jogu N. Kimani of University of M. And the next slide is the one for reference. Thank you.
Thank you very much, O.Obende. Is hoping Bruno Atamba, is he ready now?
I have not received any further slides from him. So I would suggest at this point to save time for us to close the session and break so that we can come back for the round table, because we are really behind time.
Okay, we had a Daniel Moscow.
Daniel, did I receive the slides? I did not. Okay. If you have, I don't even see him. If he's connected, fine. Not, we can close the session and come back after the break.
Okay, that's great.
Daniel is there.
Yeah, sorry, I just submitted the abstract, but then I wasn't planning to present.
No problem. All right. In that case, let's have a break and let's come and you can participate in the conversation in the roundtable discussion.
Yes, yes,
like, we will collect all the questions and then discuss them there.
Okay. All right. Thank you.
Yeah. With it, I'm so happy and delighted with my co-chair to have succeeded to come to the end of this young investigators session, which was coupled with a couple of technical difficulties. So congratulations for each of the presenters, and let us, according to the schedule, we are supposed to have a break now, a lunch break, and let us begin at 1:30. 1:30 is going to be the roundtable discussion coordinated by Professor Jose Medina Franco, and we will take all your questions there and have a conversation. So thank you very much for your time and for your presentations. Okay. Yeah, back. So welcome everyone from the lunch break. I hope you enjoy yourself. And we are going to have a closing session, which is going to be basically a roundtable discussion and closing remarks, handing of prizes, and next step of action. So I'm privileged to co-chair this session once again with Dr. Donatus Kitheka. Can you turn on your video, and then we will be glad to have you back as co-chair. And we also have, this session is going to the roundtable discussion is going to be chaired by Professor Jose Luis Medina Franco. Jose, are you there? If you're there, just turn on your camera, switch on your unmute your mic, and then we can hand over to you to coach to chair this session. Actually, it's the roundtable discussion is going to be answering some of the questions you have accumulated from yesterday and today morning, since we didn't have a chance to answer the questions. Jose, are you there? All right. We are going to have also prizes of the best investigators. The votes are going on already. And you know, someone says that in America, when the votes take place, the results are known 1 month after that. In France, the results are known the same day. In Africa, the results are known before time. Yeah, good. So anyway, the votes are going on and the votes are coming in. So we're waiting for the final. So those of you who have been contacted by email who have not yet submitted their votes, please kindly do so that it can permit us to distinguish between the very best of the best speakers, because all of you are good speakers anyway. So is Jose there? Jose Medina Franco? Yeah, if Jose is not connected, then I would say Beni and I can just go ahead and share the roundtable discussion. So please, the chats are open. Write your questions. And we are very, very, very willing to have a conversation that will last like 30 minutes. And then at 2 o'clock, we can run up the workshop. It has been 3 days of intensive work. Yes, so Dr. Eddie, I hand over to you, please, to coordinate the question and answer session.
Thank you so much. All right. Please, you want to ask a question, you may write it on the chat or you unmute yourself and ask directly.
And then some questions that accumulated from the morning, maybe we should run through the chat. Some of the questions that were posted right at the beginning of the day.
Fortunately, I don't have mine.
Okay. Sorry, that means you logged out. Anyway, let me just copy everything that is up here and repaste it. So I will just copy it and repaste it. You can follow. Don't you?
All right. Please, if you have a question to ask, just indicate. You can unmute yourself and do that directly.
Yeah, and it can also be a suggestion about way forward from here. That we don't just have another meeting and collect another certificate, but what's the way forward? What suggestions do you have after such an intensive workshop?
The participant in persons, please, you are called to do the same. Maybe you just move to Twitter, which is sharing, and ask your question directly. Do we have suggestions, the way forward, improvement, other things that did not go on well? Please help us to improve next workshop that will soon be coming after a year.
Yeah, I think it's now some people actually logging back. We see quite a number of people coming back. And we should admit that it has been quite hectic. 3 days of really hard work.
Exactly. Dr. Chigon loves who just asking if we can have some major take away message from this workshop from the host and from everyone else.
Yeah, I would say that it was first of all a major challenge to get all this together. The good thing is that we already have a template on which we're working, and every single day it's better. In terms of the presentations, I see that it was more this time around intensive in the chemoinformatics computational chemistry side compared to before. Before, the meeting was more split between bioinformatics and chemoinformatics. A lot of talks were before talking gene cluster analysis, secondary metabolite discovery, mass spectrum analysis. Like last year, we had a talk from Peter Dorison 2 years ago, and all of that. So he was trying to natural product GPS was presented, and also Joseph van der Hoof from Netherlands who gave a very nice talk the very first year. And so we have had Timan, Weber and Alex Nedema in the past. So it was more or less shared between chemoinformatics and bioinformatics. But this time around, the talks were more focused, I think, on computational chemistry for drug discovery, which we did not think that should be the case. We should say we are going to be in the next sessions a lot more open to making sure that both sides of the coin are represented in computational analysis investigations and applications in secondary metabolite discovery. That's the first observation I have this year. And the second observation I have is that there's a lot of enthusiasm from young scientists who are venturing the field from the talks that we had, particularly today, yesterday and today. We can clearly see that there's a lot of interest in computational applications in secondary metabolite discovery, some coming from developing countries, and you can see from the internet access it was quite difficult for some, but it's clear that there's a lot of interest in natural products resuscitating, and the interest is focused towards drug discovery, and there's a lot of interest in the use of open access tools, and we see a lot of web servers being used because licensed software are quite expensive, and even hardware, buying, purchasing hardware computers in the lab is quite expensive for developing countries. So you can clearly see that there's a lot of use of web servers, open access web servers like PKCSM, SwissAdMe, Warrior. There's this one I discovered today. I've never used it myself. Data Warrior. There's a lot of interest in all that in terms of also molecular docking, molecular dynamics. And so you can see that there is more the drive towards open access tools like Autodoc. We see a lot of interest in this area. So in essence, I think it's a nascent area for developing countries, and because we know for sure that resources to synthesize compounds and screen them the way any presented is quite challenging and for resource limited setting, like Stephen mentioned in his talk. And so it's so clear that many developing countries are showing interest in this, also showing interest in artificial intelligence. We saw from the talk of Gemma So which means somehow that if all these computational tools that have been used, the results generated from there be validated experimentally, then that would be a major breaking ground for drug discovery in European countries. We missed the talk today from a scientist from India, Bangladesh, or so. And so we saw that beyond just Africa, we also have a lot going on in Brazil, and we could see that the data presented by Professor Bosani, it shows quite that they're quite advanced in their data collection and physical sample collection, which means that more or less it becomes important for African scientists to begin to think about this as well. I saw from the talk of Andres this morning from Colombia and Latin America, these are biodiversity hotspots in the world, and I am thinking already that in the next sessions we should invite talks from Australia, and because this is also another huge repository for natural products, and why not North Africa? Because there's a lot from our data and ANPDB. See, there's a lot going on in Egypt, so we are really thinking in the future about North Africa, about Australia. We talked about North Africa as well, because there's a lot of representation from East Africa this time around. Yeah, so somebody said Libya. Yeah, so we that one, get ready for next time. So Libya, so there's a lot going on already, Nigeria, Morocco. So why not come on board? And we want to think that from the experience we had yesterday, where for sessions 3 and 4, actually session 3 finished way ahead of session 4. So we are already thinking that maybe we should have less talks and more hands-on. Maybe we avoid parallel sessions for hands-on and allow everybody to have a single standalone hands-on. That could be a way forward. So it's up to you guys. So what do you guys think? Should we avoid parallel hands-on sessions and have less talks and more hands-on, or more conversations, and then eventually have our young investigator forum? Because I am pretty sure that if there's more training on hands-on, then the young investigators' presentation are going to be a lot more fine-tuned. So I throw the rest to you guys. What do you think? Give your feedback on the chat. Tell me what you think. Yeah, Professor Yuta is not here. She's unavoidably absent. But of course, I'm going to channel all the feedback from her. Of course, we are going to have a survey after this meeting. And so you participate in the survey, please. And then it's after the survey, during the course of the survey, you can request your certificate of participation. So it's only after we get feedback from the survey that then we can send the certificate of participation. So it's going to be downloadable. So prepare a document that after you're done with the feedback, you can just click simply download and you write your name on the certificate and download the PDF. So that is, I don't know if there are any other things I'm very, of course, this time there was an open window that came from Eric about the fate of secondary metabolites in the environment, particularly taking into consideration the fact that countries in the tropics have different climatic conditions. And of course, with climate change, this can become a reality as well. So I've just sent out an email, and there's going to be conversation in this area as well, see how we can come together and harness our efforts towards opening this window that's coming on. So that's what I think I would say, except there are any others.
Thank you so much for that succinct summary of this workshop. And Dr. Chi is thinking that we should try to improve the curriculum by including this elementary education, in elementary education, this aspect of ethnobotany. I don't know what you think.
Yeah, ethnobotany. Unfortunately, the drive, the spirit of this series of workshops, just like we also have these book chapters, this book series that we call Chemoinformatics of Natural Products. There is the spirit and the drive behind this workshop, Computational Applications in Secondary Metabolite Discovery, is that we are looking for computational methods that will enhance the discovery of secondary metabolites, because we know that the discovery of secondary metabolites is faced with a lot of challenges. And one of them is access to data. Access to data, which is why we always encourage presentations around databases. There's access to data in terms of data for biological activity, data for the origin of the plant of the secondary metabolites, data on mass spec, like you saw today, something on mass spectrometry, data on gene clusters responsible for secondary metabolite discovery. So there is, first of all, this aspect of data. Then there's the aspect of tools. So the tools will include software, will include web servers. Then there is now applications. And generally with the application, how do you analyze all this data and how do you interpret them? Challenging for a beginner. So we bring experts in the various aspects, particularly those who have participated in developing the tools and the software. So we invite people who have developed software databases, who have developed web tools, web servers, like we always invited people from Anti-Smash. We've always invited people from, like this year we invited, actually in the previous years, actually Masha was supposed to be there, just that she was really busy. They built the database of beta compounds in Israel. So in essence, we have had to deal with inviting experts who are participated in building of these tools to give hands-on sessions and lectures on how to use the tool. So ethnobotany has not been our drive. You see what I'm saying? We quickly identified the knowledge gap and we are addressing it. Because if we want to go everywhere, it might end up being another natural products workshop, which is not the case. It's a bioinformatics or computational chemistry was natural product discovery.
Yeah, thank you so much. Dr. Siri,
Yeah, my hand is up. And I just want to tell you a little story. When I was growing up, only rich people here in Kenya had a telephone. So if you're rich, there was that telephone. So, but right now, everybody in Kenya has a telephone. I can push the same thing in a room. And even an old man, anybody can do that. So what I'm going to say is that here is an opportunity. We don't have to follow the convention. But what is important for us and what I've gained from this workshop is that AI, artificial intelligence, will make African science be a step and many steps and many years ahead. So I think I was very impressed when I think is it Jem or somebody came to Cameroon and she showed that AI can be used, and basically AI is just computational chemistry or computational analysis. So I think we should keep that in mind. That's my thought.
Thank you so much for that. That was Gemma you were referring to. That's great. Eric Home, your hand is up.
Thank you very much, and thanks very much for the organizers as well. I think it's been an eye-opener to me, and a lot of stuff was me. I think it was interesting to follow some of the questions, and I can only say that as an alumni, we're very encouraged that such work is going on there. So Fidel and the team, I would say congratulations. Wonderful organization. I just wanted to, I saw it was a lot of talk from the participants from academia, which is always good. Have you thought about industry? I mean, whether it's local or national? Maybe another. A lot of discovery work going on, and I understand it's early stage, looking for maybe target compounds or identifying compounds. Sort of coupling some form of industrial input where people from industry or their experiences, the development process, and how you could initiate contact and early contact with academia and people in industry. That's for interest from young researchers who are thinking of going into industry to understand how industry actually works, how their work is used. If you're looking for ways of broadening the outlook of next, especially local industry and international. For the local industry would be where we start asking ourselves, is there any pharma companies doing that, like that? So what you're doing, how can they look at it? Because if you look at how things work today in the pharma industry, I mean, the old days, company would take that from discovery up to market. The companies are looking for the next big thing. They don't have time to go into that area. Maybe there are things that you are looking at today which could invest in the next 10. One way to actually market this workshop. Thank you very much.
Thank you so much for that input. Prof. Tied, maybe you should react.
Yeah, it's I was just digesting what Eric is saying, and with my small experience consulting for pharma, I think that sometimes the area of interest for companies is not necessarily the area of interest for academic researchers, and more so academic researchers based in developing countries. And I think the overlap is very small, and that can be the major challenge. That's to say, for example, that scientists in academia, particularly in developing countries, want to focus on diseases that affect them most disproportionately, whether they are in South America, where there's dengue, there is all these neglected tropical diseases, or in Africa with our malaria and other things. That might not necessarily be the interest for industry. But recently, I fell on a very interesting industrial partner who seems to find interest in malaria. And then later on, when we got talking deep, we discovered that actually the project was funded by the same fund that was funding me. So in essence, what drove them was the interest of the funder. So in essence, that's where the problem is. And so we also, they were working only depending on grants. Of course, what happens when the grants run out, and it's always a question. So it becomes important, therefore, for me to say that there will be, I can propose a two-way forward. First way will be, okay, find interest in what they do and apply the skills and the know-how what you want to do. There can be a discrepancy between what you want to do and what is being done. So in essence, you can find some interest partly in what is being done, and then grab the knowledge from there, grab the tools from there, and actually in a side project, do what you really want to do. Because if I ask all of you from your honest hearts, you want to do work on neglected tropical diseases, because that is what affects your countries the most. But how about the fact that you can do this and this can never be carried to the market? So Eric's idea would be like, do something that is already working and invite others on stage to take it over.
That's right. That's right.
So that eventually Twitter can become X.
Just to add that tropical and infectious disease, that's a big topic in industry today, whether in the international pharma industry, the malaria, because they are not going anywhere. So fire, there's cancer, there's all this that's going on. It's still very big in international pharma companies. I don't prefer to be an expert in this issue, but I know industry is looking for just this infectious disease because it's something that is affecting very, very big population of the world. It's not, I mean, cancer is important. If you look at the proportion of people who have malaria or tuberculosis or all of this, the population, so industry that is financially as well. It's not being neglected, I would say. And the best way is to sometimes not just very difficult to go into industry and say we have this. So just somebody from industry to tell just a 30-minute talk here, just to say how can we potentially use what we're doing in our work, or how do we do our own work, just to as an eye opener to early researchers to know that, because there's one thing academia industry is quite a big different world. Just to start introducing this concept, I mean, people may be coming to the end of their PhDs or their master's degree, a lot of work that they have done. They may want to do something, not yeah. So what do you do with this work that you've done? That's another way.
Yeah, sure.
And just to add that, I think even the models that people have done, I think I've been just looking at it the past few days. It's quite an interesting software that, I mean, it's not, I thought that there are outputs there as well. So it's something that could be important, not just for drug design. I mean, there are compounds that nobody has data or that you put in there and see what is predicted. And I mean, sometimes in the absence of no data, even predicted information is important. So those models, they're not as, they are important as well. Well, nice to get to know that model. I think I have a to that now. I spoke to the man who took a look at so this work, sometimes nobody knows where they are. If I didn't attend this, I will not know of that work. So it's a lot of work going that it's not actually out there. So market this as well. Not fair.
So actually, Eric, what you're saying is very pertinent. I would say, for example, Yannick Jumbo-Fenan works for an agrochem, and he has been on this from the very onset. He is very skilled chemoinformatician, bioinformatician, very skilled. He has worked on one of the big projects you can think about, Human Genome Project. He's participated in PopChem development. He's been involved in many projects, actually, originally from Cameroon and now based in the United States. To get him to speak this time was quite challenging, even to write a chapter in the book series we're talking about, because he has to go through a number of ticks from his hierarchy to be allowed to give a talk. So eventually, until this workshop, he was not. I know you have clearance, but his immediate boss says, in that case, you can present as an independent person. So you can see some of the challenges.
Yeah, I mean, I will feel that sort of challenge if I had to invite a colleague of mine. There is no harm in trying.
Yeah, so we can always have to depend on people like you who are inside, because I'm told in the proverb, an African proverb that says that salt only gives value when it's inside the soup. So when you are in the soup, you can always give us more value. So in essence, this is a nice opportunity to bridge academia and industry. We also have the chance to work with people like Celia, who are not really a company per se, who are non-for-profit, who are just rising. So we are more or less gaining from them and all. So,
yeah, thank you so much. Dr. Chi is buying your point or suggesting that the hands-on sessions maybe should be arranged so that everyone can participate, all the sessions.
Yeah, it can be a good one as well.
Yeah. And Angela Marcaye asking if let me just read what she has written in the chat. I'm getting trouble on how to analyze data, especially on empty simulation results. Is there any possibility of including analysis of data in the coming workshops for training, Prof?
Yes, that's a good question, actually. I read it already, and I'm really excited to say that people are doing some things already. The first quick answer is that yes. The second quick answer is no. And they said yes, in the sense that if you have data already generated for MD, it's easy to show you how to analyze it. But if you have to run the MD during a hands-on simulation, hands-on session, it's not feasible, because some MD will take 2-3 days, from my experience. And so if you start a hands-on session and you want to last for 90 minutes, get your data and analyze it will not be feasible. So, which means that if we already had pre-run data, it will be easy to show you how this works. That's the first thing. The second thing is, then you ask me, how do I generate the data? See, so in essence, I would say that that it has always been our intention in the hands-on session with Daniel to do a complete virtual screening workflow, complete virtual screening cascade from the start to finish. And what we usually do when I go to Tanzania is that since they have a 3 weeks workshop, Tanzania, so if you have a chance to be part of this workshop, in-person workshop with Daniel, also Professor Soliman in Egypt, also in South Africa, in KwaZulu-Natal, also runs a workshop on molecular dynamics. So Soliman, that's the name, and he runs a workshop. I can always give you his contact if you want to participate in hands-on focus on MD. So when I attend the meeting with Daniel, I usually teach visual screening from pharmacophore-based visual screening. And it's really fast and quick. You can get your data very quick and you can be able to analyze it. Now, when it comes to molecular docking, it's also relatively quick. Therefore, MD is quite challenging because, the MD simulation is computationally expensive. What's still metadynamics? So what we usually do is that the student, the first days, prepare their files and do their first runs. While the computation is going on, they are now attending other courses and doing other exercises. Then when the results are out, they now be able to start analyzing it. So it doesn't take place in 90 minutes like this one. It runs over 3 weeks. So this kind of workshop is just to give you interest to throw the corn, as they say in Africa, but throw the corn for the chicken to come. Then if you want to really put your hands on MD, the first thing you should have in mind is that you must have access to a cluster that can run these calculations because you cannot run it on your laptop or on a simple desktop computer. The second thing you must have in mind that it takes time, so which means that you would want to really attend a real-time workshop, hands-on, in person, which will allow you enough time to get the result and analyze it. So I don't know if my answers are satisfactory.
Angela has unmuted herself. Maybe she wants to say something. If not, Dr. Steven suggests that we should have young scientists or presenters from all the corners of Africa, the north, south, west, east, and central.
Yeah, it's an ideal situation, actually. It's an ideal situation. What we do is we prepare the flyer. Like now, I have a list of those who attended this year, which means that I collect that list and invite systematically everybody on that list, all the speakers from the past years together with the participants. They are all invited. Sometimes I get emails bouncing that people are not going to respond to those emails. Then the second way we do it is that we also get our posters advertised and people register with abstracts. So we actually collect. This year, we did not turn down any abstract, actually, except those who were not available and those who, for technical reasons, were not able to participate. So that's the thing. So we get as many as are willing to participate, and we try to create opportunities for them. The young investigators who don't find a space to give it a talk during the young investigators session, are often we put them in the normal oral presentation session. For example, some of the young scientists actually spoke yesterday. So in essence, we actually are encouraging many young investigators because it's a way of gaining experience. It's a way of learning a lot of things. And so this year, for example, in my own group, only 2 young investigators were able to present, whereas there are other people who could actually present because we give preference to others. So that's it. So we are really, this idea is really welcome, and we will actually want to invite more persons to be part of the organizing committee beyond just myself and Professor Ludwig Miller. So we would like preferably for next year to expand our organizing committee, have a real scientific committee, a real local organizing committee, so that things really take their shape like a normal workshop.
Thank you so much. I think we are at 6 minutes past 2, and I'm not seeing any other questions or remarks or suggestions.
Yeah, thank you very much for co-chairing this session with me by impromptu. I just checked now, and I have not yet had any further votes coming in for the oral presentation We have come to the end of our this year's computational applications in secondary metabolite discovery. From the previous years, we're encouraged to have it every other 2 years. So we are going to decide now, do we want it next year or do we want it in 2026? Usually it's in March, the early part of the year, first quarter of the year, either late February or early March. You guys prefer 2025? Yeah, somebody said 2025. Yes, right on the text. 2025, 2026. Yeah, another person 2025. How do you get certificates? We are going to send a link for you to complete a survey. In completing the survey, you are going to request for certificates. Then you download it directly PDFs. If you are not able to get a link within 1 week, get to me. And everybody else is saying 2025. All right. That means we validate 2025, and so we get ready to work next year to have another workshop and more expanded. And this time around, avoid parallel hands-on sessions. And preferably, actually, I was even saying people come with their laptops ready to, and the hands-on people send their files way ahead of time, so that while they are doing it, others can also be participating at the same time. All right. Everybody else says 2025, so it's noted. Local organizing committee is noted 2025. It's also noted that we don't have parallel sessions. Hands-on sessions are done one at a time. Yeah. And they expect to have their survey and complete it and request for your certificates. Now I must read out the votes I received so far. I don't know if you are there, but we're going to send out the certificates for the best 3 oral presentation. Unfortunately, we cannot have a cash prize because the cost of transferring money around the world is more than the cost of the money itself. Okay, so I have received the votes, and I hope you guys are clear. All right, based on the votes I have here, it was unanimously decided that the best oral presentation from Young Investigators Forum is from Dr. Donatus Bekindaka-Eni. I expect a clap and applause for that. Yeah, that is, I can expect the in-person people are actually clapping already. Then that is it. Then the second, there is actually, I would say 2 seconds that were brackets. There is Hannah Augustijn from Wageningen University was voted as the second best presentation. Alongside, she's paired alongside with Evegenii Ziaikin. Evegenii Ziaikin, both Hannah and Evegenii Ziaikin were second position for Best Oral Young Investigators Award. Okay, so those were the 2 second positions. And the third position, that Best Oral Presentation for Young Investigators goes to Mr. Betow Jude Evegenii. Those are the awards. You are going to have your certificates. Please request for your certificates as evidence that you won this award. As a young scientist, it goes to your CV. And congratulations, and looking forward to you guys on behalf of Professor Lili Mula and myself to call computational applications in secondary metabolite discovery 2024 has come to an end. Have a good time, and let's see each other next year. So I leave the forum open, and those who want to continue talking can talk. I mean, allow you about 1 hour more. Those who want to exchange, contacts. Congratulations to those who won the awards, and for those who want to exchange contacts, the forum is open. The forum is open. All along, I've been sending individual emails. Congratulations to all the winners, and wishing you all the best. You are going to have your certificates, and that will be it. Oh, congratulations. All right, all the best.
Dr. Eni.
It's on.
Are you on? Yeah, I was impressed by your presentation.
Thank you so much.
Yeah, are you? I mean, have you thought about using diversity-oriented synthesis of natural products?
That is going to be a good one. I have not thought of that.
Yeah, I have a student, a master student working on such a project, and I think it would be really nice to use natural product scaffolds to get into some different chemical species. Just using simple chemistry, not necessarily those organometallic scaffolds, just normal simple heterocyclics, like using the Fischer indole syntheses, and I think that you just what do you do? You just reflux, just reflux reactions. So if you're interested, it would be really nice to pursue that. And then we also convince Fidel to join us, especially in the application of artificial intelligence in that area.
Yeah,
So,
I am open to that. Thank you so much.
Okay, brilliant. Yeah. So just get my email and just get in touch. We can put something out.
Okay. Please just send it in the chat. Let me take it down.
Yeah, that's fine.
Oh, thanks so much.
Okay, sure. So let's just keep in touch. And you see, just I'm out of applying natural products chemistry and making it easier, more interesting. It's good. Yeah. Thanks for your presentation. It was,
Thank you so much.
A real honor. Thanks.